Hi everyone. For those new and just tuning in, welcome. And for those returning, welcome back. We have quite the workshop planned out for today, and we're very excited to be showing you. So again, welcome to the Carboncopies Foundation June 2021 workshop event. This one's titled ELEGANS, the First Emulation. So my name is Bill Nguyen, and I will be helping to moderate for today. And as a reminder, this event is produced entirely online with the generous help from our donors, our patrons, and our team of volunteers. Now, today's event in particular is topical and actually deviates a bit from our roadmap update series, for those familiar. And for this event, we'll be discussing the nematode species C. elegans, and in particular, its significance in neural data collection research, as well as in behavioral modeling. Here's how we sort of anticipate the schedule working for today. To start off, we'll have a brief video introducing the topic. At around 8:15 a.m. Pacific time, we'll stream our first interview recording. This first interview is with Dr. Stephen Larson, who is the CEO of Metacell, as well as the co-founder of Openworm. At around 9:15 a.m. Pacific time, we'll start our second interview, this time with Dr. Natasha Vidamore, who is the Executive Director of Humanity Plus. And at around 10 a.m. Pacific time, we'll start our live panel discussion, and wrapping it all up at around 12 p.m. Pacific time, Dr. Randall Kuna will close off the session with a few summarizing remarks. So now I'm just going to go over a few technical comments regarding how we sort of expect the workshop to work. So in my best narrator voice, our workshop welcomes participation and is open to live participation by all of our international attendees and viewers. For viewers on YouTube, you are invited to write questions directly into the chat that is enabled for our YouTube live stream. One of our excellent staff members is monitoring the chat and will alert the panel to interesting and topical questions. Longer or more complicated questions where it is important to establish that the question is both properly understood and answered are best asked by directly engaging with the panelists. We make this possible through our Callin Number. The number and PIN are shown at the bottom of the screen right now. They're also written in the live stream description and on the livestream.carboncopies.org webpage. They are as follows: 786-540-5631 with PIN 180-618-986#. If you're calling from outside the US, you may also use the web call-in option at call.carboncopies.org to avoid telephone charges. Now, this process is similar to calling into a radio show. Your call will be monitored and a respectful exchange is expected. When your question would benefit from direct interaction, I'll invite you to call in if you can. If you want to add a note to your question stating that you can call in or that you can't, that can be very helpful. Now, before we get started with the first interview, here are a few additional technical comments that are important for our panelists and for others who have joined us directly through our online meeting tool. So as a reminder, please mute your microphone when you're not speaking. If you happen to also have the YouTube live stream open on your computer, please mute that YouTube player to avoid echo effects. We'll be presenting our recorded interviews through the online meeting tool. And unlike some of our earlier workshops, there is no need to switch from the meeting tool to YouTube to watch the interview recordings. And please let me know if any of this is unclear or if you experience any technical difficulties. I also want to take this time to remind everyone that we are very thankful to our patrons and donors, and especially to our team of volunteers and the many experts who sort of take time out of their busy schedules to help us out with these workshops and allow them to be possible. And as a reminder, we are a 501c3 nonprofit operating with the sole mission of advancing research towards whole brain emulation. As a result, we appreciate the many contributions that have let us set up events such as this one. And if you want to become a monthly donor or make a one-time donation, we have a page set up on our website, carboncopies.org/donate. You can donate either by PayPal, credit card, bank transfer, or cryptocurrency. And we greatly appreciate your help in helping set this up, whether it be through volunteering your time or through financial donation. With all of that now addressed, we present a brief video sort of introducing the topic for today's workshop. Nutrients and bacteria. C. elegans, or Caenorhabditis elegans, is a transparent nematode worm about 1 millimeter in length, found in soil within nutrient and bacteria-rich environments. Members of the species have a lifespan approximating 2 to 3 weeks. Its neurons contain dendrites that extend from the cell to receiving neurotransmitters and to the worm's nerve ring, which is essentially its brain. Moreover, C. elegans has motor excitatory and inhibitory neurons acting as regulation for the worm. Collecting data using calcium imaging from the C. elegans brain is easier compared to other species, both due to the worm's transparency and because it only has 302 neurons. Because of this, the C. elegans serves as an efficient research subject. Currently, there are hundreds of labs using C. elegans for research, each lab specializing in a different focus area. In today's workshop, we will be discussing the research and modeling methods of the C. elegans and explore the next challenges to be addressed. Okay, now before we proceed with the first interview, Dr. Kuna, do you have any introductory words?
Yes, thank you very much, Bill, for that wonderful introduction. I want to make sure, before I even get into a quick introduction of how this all fits into our work, I would like to just take a moment to say very few administrative things. And one of them is directed at all of us team members and guests who are participating live in our Meet channel that we're using for this call. Because the chat window tends to show whatever anyone is typing in there, it's not the best way to have conversations, say, behind the scenes. If you find something interesting or you want to make note of something, in that case, it's better to use what we call the back channel document. I think it would be great if one of our team members, Michael maybe, or Thomas or Bill, if you can send around to the core team, if you can send the link to the back channel doc so that people can use that instead of the chat. That's just a point, because in previous times we've had things pop up in the chat while streaming. Now, okay, there's, I guess, one other thing that I wanted to mention is that it would be great if there is someone checking to make sure that the live stream on YouTube is always in good order. I hope that you guys have arranged that already amongst one another in the events team. I just want to check because we have, of course, sometimes had issues where the stream would suddenly drop or something like that. Now, as to the topic, it may seem to come a little bit out of the blue that we're suddenly talking about the nematode worm to some of you, and others may think, oh, of course it's the nematode. We have done a series of workshops now, a long series that's been going on for, I don't know, has it been more than a year maybe? It's the series where we're trying to gather the information from experts to do the update on the roadmap, the update which, by the way, is proceeding nicely and will hopefully soon appear as a beautiful wiki-style, approachable living document. And so we've done this collection, and as we've been going along, we've started to enter the domain where we're talking about, okay, in a realistic way, when you collect the data from the connectome of the brain, so that was a chat text, but I guess that was for the back channel document, so that should be okay. When you take the data of the connectome, whether it's by, say, I don't know, some kind of vitrification or other process of preserving a brain and then imaging it. And then you rebuild the brain, that three-dimensional image, and you try to transfer that into a functional model. It's a really big problem. It's one of the most difficult things of the entire process, really, probably the most difficult thing. And mammalian brains are just very big and very complicated. And we know very little about that process, how to make that transfer from what is basically a three-dimensional image of the connectome to a working model. And we've discussed this transition a lot in the context of mammalian and human brains in the last few workshops. But to try to learn a little bit about the methods and to get to the point where you can actually look at that transfer happening and see what works and what doesn't work, you have to step back and look at very small, tiny brains. And this is one of the smallest. There are some unique aspects about this brain. There are some things that are quite different than what you would find in a mammalian brain, and I think this will all come up in the conversation. And that's maybe one of the reasons why many neuroscientists don't study C. elegans. But I think it's still very useful just to see how in this very small animal, how you actually make that transition, how you can connect those two areas, that mapping of the connectome and a model that actually works, something that is an emulation of the worm. And that's the reason why we decided to do this. I wouldn't even call it a detour. It's kind of like this look at this adjacent territory that gives us really useful information about how those methods are going to work. So with that, I hope you enjoy the talks that our guest speakers are going to give, and then the ensuing conversation or debate.
Thank you. At this time, we present our interview with Dr. Stephen Larson as conducted by Dr. Kuna.
Dr. Stephen Larson on C. elegans. Dr. Stephen Larson is the CEO of software company Metacell and co-founder of the worldwide Openworm project. Dr. Larson has presented and published about C. elegans research in numerous prominent forums and academic journals. In the following interview, conducted by Dr. Randall Kuna, Dr. Stephen Larson describes why the nematode worm captured his research interest and the applications of that work for neural network data collection and simulation.
No echo. All right, so the reason why we asked for you, you know that our organization is interested in whole brain emulation, and it's primarily about human whole brain emulation, so the human brain or mammalian brain. And for that, it's extremely important to be able to obtain the connectome data, reconstruct it, and functionalize it by mapping from, what's basically just a 3D image to something that is a functional model. And as you can imagine and probably know, working with mammalian connectomes is enormously difficult because they're so huge. It's very hard to obtain a good connectome. Even the cubic millimeter petascale work that Allen Institute did, it's still questionable exactly how good the results are. Some of the people who've looked at the first slices there, the images, say that maybe they're not all as sharp as they could have been, so then reconstructing can be a little more difficult. So it's just very difficult to obtain good data and then to extract something useful from it, because hardly anyone's done work in that. It is almost impossible to do the kind of simultaneous studies where you have, say, a connectome and you also have the functional system, maybe with calcium imaging. I mean, you could do that with zebrafish larvae or something like that, but then you're already outside of the domain of mammals anyway, right? So it's kind of like, it's hard to do with mammals. And then there are special cases, problems like how do we identify gap junctions and kind of map them to types and responses and all that. So plenty of problems there, and that's why small animals are really interesting. And of course, that includes not just C. elegans, but also Drosophila, and then maybe a few other things. There's the mollusk and that sort of stuff. But that's why we're coming to you, because Openworm and C. elegans as a whole is just a field that seems closer to results of some kind. But what led you to C. elegans. How did you ever get started? What made you decide, oh, nematode work? This is awesome.
Indeed. Well, and I'm curious, the macro, I'm happy to, of course, answer your questions, but also the macro strategy, because I believe, I spoke with, I did another interview with someone in your cohort maybe 4 or 5 months ago, right? It's Randall? Am I right about that?
Was this an interview for the strategy project?
Or
it could be
some other person.
Because I know I've spoken with you before. We've had an interview before, but that was a long time ago. But a few months ago, I think you were approached by a member of our strategy project, because we have had a conversation, but it was a bit about way back. So I don't know. Michael, do you remember who it was that reached out from the strategy project? Because I do know they did their own interviews.
Yes, the strategy project.
Yeah, I'm sorry. At Carboncopies, we've gotten to the point where the organization is big enough that we've had to split up a lot of responsibilities, and that means that even though I'm aware of the big picture and how it all fits in, small details like who did what interview when to collect the data or whatever, that may have kind of slipped.
That's okay. Actually, every time I talk with you guys, I enjoy it because it also stimulates me and it gives me ideas. So I'm happy to chat with you guys often, in fact. But I was just sort of losing track of where you guys were in terms of, because we were talking a bit about the whole brain emulation roadmap, and it was in need of some revision, and there was this big idea to take a look at where it's come, and it's been some number of years, and so we sort of got into some Openworm stuff. Again, very happy to talk about it, but just in case.
And that's all in the works. That's all very much under construction right now. There's a wiki being created where basically everything from the old roadmap is being redone and ported over. New research being put in. It might also interest you to know that I'm working with Voxa right now. So they're the team that made the automated microscopes for Allen Institute to collect that cubic millimeter. So I'm deep into connectome stuff every day at the moment.
Nice.
but it's more on the technical side of how do you make it collect properly, and that's why things like is the focus good or something like that. Yeah.
So, but then I'm just wondering if this is not a follow up to that. So this is kind of a
not directly. No.
Now I'm talking to Randall, which of course you're a very strategic person in the organization yourself. So I just was wondering, what are the larger goals of this chat relative to that one? Again, not because I'm
the larger goals are that we've had a number of workshops now where we've been adding in information for this new roadmap, and some of the most recent workshops have been about connectomics. We've sort of gone down that, mostly about once you've collected, electron microscope images or something like that, how do you get from there to a working model? How do you translate that into something that is functional?
Sure.
And it seemed like a good moment to get away from human brain and to go into simple, smaller animals. And simple is relative, of course, but much, much smaller than the human brain. Just to get that perspective and to understand what's possible even at that scale. And so that fits into the strategy of how do you tackle problems like going from connectome to function. And I know that the approach, the strategies can be different for a small animal like that than for something like human, but still it's good to learn. And so we're just trying to collect some information. So this belongs into our pile of the workshops that we're doing to collect knowledge that we then put into this revamp of the roadmap.
Cool. Got it. Okay. Thank you. So yes, happy to dive in with you. So how did I come to do nematode work?
Yeah.
So I think it was out of running into a series of roadblocks. Okay. So I started I started off as a computer scientist. I studied with Patrick Henry Winston at MIT, the late and great Patrick Henry Winston, who led the MIT AI lab after Minsky and was a student of Minsky's, and who was firmly in the symbolic systems camp, which of course was sort of going out of favor. My master's thesis with Professor Winston was trying to convince him that it was okay to take one step into connectionism from symbols and find a way to kind of bridge the symbols approach with connectionism using self-organizing maps. So he actually, he was a very nice guy, but I think what he thought was good work, it was hard to convince him, but he thought this was good work. And the last thing that I did that he thought was good work, by the way, when I came back to him with the nematode stuff, he was like, "Well, I still think your thesis was better than this. I don't get what you're doing with it, but be what it may." So, but he was all about biologically inspired AI. That was his thing. He was very much hardcore, looking for AGI. Didn't really label it that way, but he was, interested in the way the humans told stories, how humans understood stories, right? And so very much like that was his perspective on human intelligence, is that, you should be able to build something, should be able to understand the story, understand who's in there, the roles, the characters, be able to have conversations about stories as written. But his focus on biological AI led me to reading more and more neuroscience papers and understanding cognitive science as much as I could. And that led me to, after a while, trying to very poorly attach cognitive theories and hypotheses to the anatomy of the human brain. It led me to actually want to study what was known on the biology side of the human brain, because I was ending up spending a lot of time and money, like just consuming textbooks and what I could understand from the published literature of neuroscience, which at the time, before a graduate degree, it was limited. I had a very limited ability to understand it. So went to grad school, got there, came in with this AI perspective, got to a neuroscience program through an engineering on-ramp without a biology background before that, and learned that through the coursework, and kind of landed in looking at rodents. So taking it one step down from humans, but starting with rodents because there was more data, and it was very quickly apparent that making computational representations of the tissue itself would be an important foundation to just set down and sort of say, like, okay, well, what are the tissue anyway that we're attaching these cognitive theories to? Like when we say cortex, when we say thalamus, when we say cerebellum, what does that really mean one level down at sort of the epistemological level? Like what is the formalisms that underpin the anatomy? Because if we don't have a formal way of talking about anatomy, I thought it would be difficult for us to ascribe things like memory and reasoning and like all those other human things to the tissue itself. And of course, what I discovered was that there wasn't a very formalized approach to anatomy in the literature. There were a lot of problems, especially when you try to reduce it to formal logic. So it became very problematic. And not only that, there's multiple scales to deal with. And we care very much about the level between sort of cells and tissues, the circuits level. And so bridging those required also bridging literatures. Turns out the researchers in these areas have different methods. So my PhD work ended up being mainly, trying to teach a computer anatomy, neuroanatomy. And after doing a lot of work in the cerebellum, the supposedly simplest part of rodent anatomy, which of course also not simple, but because of folks like Ito, who had written some very compelling simpler descriptions, seemed addressable. But that just opened my eyes to how big the information management problem is when it comes to this project of laying down a foundation of anatomy upon which you can ascribe cognitive functions, that sort of thing. So of course then, things like whole brain emulation, where you guys are looking, at a very tissue-centered way, is very much consonant with where I was coming from with this. So basically, again, I said, how did I get to C. elegans? By hitting a series of roadblocks. Well, because I wasn't able to, do a good job of ascribing cognitive function to tissue, I had to learn about the tissue. Then because I wasn't able to do a very good job even dealing with the tissue because it was still itself under constrained, it was my strategic retreat, essentially, to go to an organism where, we actually had the right size of organism and the right quantity of entities relative to the problem of formalization. So that's really why. And it seemed to me that the promise of figuring out some tools at this scale that you could then bring back up the ladder of model organisms would eventually be the path that research would take. And it had happened several other times for like the molecular markers, like GFP basically started off in worms, sequencing started off in worms, and lots of stuff, started off in worms first. So so that's how I got to C. elegans, by a desire to find the right epistemological level of description where we could assign function to cellular activity and just strategically retreating back down the evolutionary scale, if you will.
Okay, yeah, that totally makes sense. And of course, we've all experienced this, this sort of ladder and then going back down the ladder to try to find something. It's actually a problem you can tackle, that you can constrain enough that you can do something with it, right? And it goes even further than just going to C. elegans, I suppose, right? Then you have to decide which part of that problem you want to tackle first, what data you can actually acquire and how you can map data to parameters. Because the problem is, of course, always that when you do modeling, you never measure the parameters you're trying to put in your model. You always measure something else. You're always recording some other data, and then you have to transform it into the parameters that actually matter to your model. And so there's a lot of iteration there. So in that sense, then, even though you ended up at C. elegans, I guess, I'm just guessing, you can tell me if it's true, from when you started to now, you probably also ended up going through a giant learning curve on what works, how, and what the big hurdles are. What would you say?
What I say was the learning curve about what works in the area of C. elegans. Yeah. Right. Well, so now, having arrived at a place where you could start to formalize, right? We did a lot of databasing that we couldn't do before. And so we could identify individual cells. Now we could describe circuits. We had a connectome. We could throw a connectome into a simulation engine that was the entire thing. We could flip it on and start to see what happened. And we had the advantage of very tightly controlled behavior. And even folks doing, sort of whole nervous system recording, calcium imaging, right? Mm-hmm. So what you start to realize is what some of the limits are of the, experimental to theoretical exchange that are possible, right? And so there's a lot of just starts to be practical problems of where I think where the project has kind of gone is we're still, I think, tied up honestly on some of the logistical things. I think that it's still the right space scientifically, but now because the population of people that do this work is very small, and they're kind of in the particular institutions they are, and they're doing the particular research they are. I think one of the challenges has now become how to convene the right interdisciplinary conversation, basically, with this very small number of people with very specific scientific skills. And we're talking about like handfuls of labs now in the world, most of which I know, but they have their own agendas that are animated by, things in the academic space. So, for example, right, the best labs at the movement quantification basically is like one or two labs. Then but they're different than the labs that are doing the really detailed calcium imaging work. And they're not particularly interested in combining methods because they're on a path. These two labs, but they absolutely need to combine methodologies to populate a data set for this model. And then neither of those two labs are expert in simulation methods. And the simulation methods that one requires for this kind of work is like computational physics kind of work. And so they're also disconnected. And then the groups that are the best in computational neuroscience also don't really talk to these. And then there's a need for folks that do sort of single cell work, which is like a particular group that's best at this is out in Stanford. So here, I think over the last 10 years, I think the challenge has been recognizing that some of the unfortunately practical funding limitations and practical inability to pull everybody together to sing on the same page as much as we have done in Openworm, but getting them to actually be on one team doing one project to simulate this thing, basically to like build the Allen Institute for C. elegans, right, would be the approach. To bring everybody on site and give them one mission, right, is probably the thing that has to happen next, I think, in the community in order to make real progress. Because I think all the tools and the elements are there, but because of like a limit of how many cats you can herd when you start to want to do serious research in the space, not only that, even to convince a funding body to fund it in an unconventional way, instead of like, what do you mean I'm not just going to give all my money to one university? Or what do you mean I'm not just going to give my money to 3 PIs and they'll work it out, which they generally don't. We've found. So, but yeah, I think to in the scientific space though, let me to make it not a logistical thing. What are the questions?
Is extremely important. I'm not complaining about discussions about logistics.
Well, yes, and we can talk about that. But let me give you more insight on the scientific space. So what becomes interesting about the worm, right, is the proportion of neurons which are the most analogous to the human brain relative to the proportion of neurons that are analogous to our sensors, the things that take information from the world, and our effectors, the motor neurons that are directly attached to the muscles, are very different, right? So in humans, of course, our central nervous system is so well developed that, I don't know what the exact fraction is, but like 80–90% of the neurons are in the central nervous system. But in the worm, it's more like 40%, 50% are the central neurons. So what's really attractive about that is when you're dealing with 300, on the order of 300 neurons, you can start to very quickly isolate down to the neurons where the surprising stuff is happening, where I think the algorithm that we care about, the biological algorithm that we care about is occurring is about 70 neurons. And you can kind of pin down the activity of the sensors and the effectors into kind of a lower dimensional subspace and kind of be like, I think I kind of know what's going on with the inputs, and I think I kind of know what's going on with the outputs. Certainly to, one synapse up from the muscles, it's like we kind of, I think in Openworm, like have a pretty good guess at more or less what they should do. And, the sensors are kind of themselves like cells that are specialized to transduce information, from the world. So they're up to the first synapse in from the sensory neurons mostly. You can kind of know what they're doing. So what that means is I think the interesting stuff is now a very small number of neurons total. Again, on the order of like maybe 70 to 120, depending on how you carve them up. And so then you start asking, well, okay, what's special about what those neurons are doing? And I think what's the most interesting for me is if you think about evolution took a long time to build nervous systems, relatively speaking, at an evolutionary time. And it probably arrived at some patterns to make nervous systems do what they do. And then, as evolution likes to do, it probably iterated and built upon those patterns, right? Yeah. Reusing things that worked in the long period where it was taking its time to evolve. So what we've been pursuing in Openworm, if you get right down to it, in the 10 years it's been there, is to say, yeah, actually, we want to isolate down to, we want to look at these interneurons specifically, kind of control for what the inputs are, kind of control for what the outputs are, more or less, and focus in on, well, what's special about the dynamics in the interneurons that might be an evolutionary key that unlocks what other nervous systems that are larger and more articulated, do in some way where there might be a pattern that we could, the gold mine is, can we abstract an algorithm that we could define in a different way than just the gyrations of these particular neurons, right? Something that we can say, like in the, the David Marr, like the different levels of description, like algorithmically, implementational. Do you know what I'm talking about, Randall? Suddenly I can't hear you though. I think you muted. I think you're talking while muted.
Oh, sorry. I muted myself because there was a plane coming by and then I forgot to unmute. Yeah, no, I know David Marr's model with the 4 different levels and yeah.
Right.
Yeah, sure. Yep.
So the gold mine, of course, and this would be the same. I'm sure it's very, I'm sure this isn't a roadmap, I'm familiar with it, but this thing about like abstracting one level up from, simulating the neurons to unpacking the algorithm and being able to define that algorithm in any other way that's not the physics, right, is the goal. So so lately, that's been the highest area of interest, and we still have to simulate in order to basically, the simulation needs to match the data, and then the models, the higher level models, can match the simulation, so that then you have a way to check to see if you're right, or to see if your more abstract algorithm is a very small subset of what the dynamics are actually carrying out. There you go. There was a lot of stuff. I don't know.
Yeah, that's a lot of stuff. So would you say that that there is a lot of resemblance, that you can look at the interneurons in this set of 70 to 120 cells and say, these interneurons are performing a similar function as a network of interneurons creating a certain modulation or something like that, or is it kind of flipped around where, say, interneurons are doing work that normally principal cells are doing? How different is this system when we look at it for people who, like myself, who really don't have any insight into C. elegans, and we don't know how similar or different it is to mammalian brain, except for, we've all been told, well, there are no populations, so every neuron basically does the function of an entire region and sort of regionally important. It has its very unique specialization or something, but that may be a mischaracterization too.
Yeah, it's, I think that could be a very productive conversation if you and I together can collaborate to, I'm just in this conversation, to like find the right metaphor here, because I don't think it's like one neuron to one brain region. That's the wrong mapping function, basically. So these neurons, I call them interneurons, right? But I know, right, in the central nervous system, you think of interneuron in contrast to a principal cell. But in this case, but in the central nervous system, principal neurons and interneurons are all contained within the central nervous system. And here, this is the central nervous system of the worm. So it's all in, mostly all these neurons occupy what's called the nerve ring, which is sort of located up in the head of the worm. And it's literally a ring of neurons that goes around the sort of the mouth muscle called the pharynx. So they are, first of all, highly recurrent. So they're all synapsing on each other, gap junctions and chemical synapses. Some are excitatory, some are inhibitory. In terms of specialization, a lot of the neurons in this region have not been ascribed single functions. If you really start looking at it, obviously in the worm, people like to ascribe functions, but if you dig into it, it's like, oh, this is sort of a motor cell, because maybe it's been correlated with some movement, or this has both motor and sensory activity. So I think what I've been looking at is more how they operate collectively. And some of the papers that have been coming out where they've been doing the calcium imaging on the nerve ring is revealing that they are much more, their activities are more correlated with each other than they are uncorrelated with each other, meaning that they're operating as a functional unit, but they are potentially even operating in like a hierarchical manner. A recent paper last year was suggesting, but they're doing it as part of a team, as if it's not that individual neurons have specific functions, but the fact that they're clustering together, they're creating a functional unit that is more than the sum of its parts, if that makes sense.
Yeah, sure, it makes sense. And I'm not surprised that the sort of this sort of, just saying, oh well, every neuron in C. elegans has a specific function, that's not entirely true, because it wouldn't make sense. That would be a waste of how you would use a resource in a network like that. And that's why, there's this tendency, people like to do that. We like to call something a Jennifer Aniston neuron, but that's silly, because of course it's not only doing Jennifer Aniston images, and it's not only involved in that, and there are other neurons involved in the same process. But yeah, it's a sexy thing to do, and you get publicity and papers and all that sort of thing. But yeah, so this kind of leads me back to wondering about what the strategic hurdles really are, because from your description, it does seem like a very interesting and more tractable problem to deal with. So I kind of wonder, why isn't there a large project to do this? Why is the set of people working in that field so small? Why are there only a few labs? And what is it that, what are actually, let's say, what are the reasons why there isn't just, say, a version of an emulation of C. elegans that is really rough, that people can say, look, this is number one, this is actually an emulation of the thing, and sure, it's not perfect, but now we can improve, where people can just point to it and say, see, the first thing has been done. And it's just, okay, it's crappy, but it's there. Why can't we say that exists, or does it?
So it sort of does. But it's currently, so, when we've had this vision in the project to make something that's very accessible to people. And along the way, I think we had a crossroads for what it took to build the very accessible, user interface, if you will, to the C. elegans model, versus what it took to do something that we could publish papers about. And we chose the route in the medium term to do the latter. So what exists right now, we've written about in 2018 special issue of the Philosophical Transactions of the Royal Society. This is kind of our big publication push that we did now a bunch of years back. It feels like it just blinked, and it was fall of 2018, but obviously time has moved on. So what we have built exists, and we just recently, I think, released a new version in the last 6 months at github.com/openworm/openworm. But it is not the sexy thing that is very highly easy to use. One needs to be kind of a command line programmer to be able to run it. But what is there is a Docker container that pulls in specific working versions of code from around Openworm, where we have our modeling package, the computational physics package, the computational neuroscience package, and it pulls in some data sets. And if you download it and you run it, it will run, well, by default, it'll run a very small chunk of the simulation of the worm. If you add some parameters, it will run more of it. But it is able to be run on sort of any computer that can, access Docker and has enough space and all those sorts of things. I believe I personally run it on my Mac about 2 months ago. I believe it works on Windows. I have not tested it myself, but I think some Windows folks came along and did it. So it's a starting point, but I think, yeah, I think the thing is, we still need more work and effort to do the more sexy PR push, probably, to point people at it. Not to mention the like, actually the goal of getting people to understand what's in there, requires a lot of dedication, I'd say, at this point. You need to kind of work out, okay, which are the repos, and okay, go read that. And we've been reworking our documentation and stuff like this. So, but nonetheless, it represents kind of the last decade of work that we put into it. And it is also a foundation, with the nice thing about technology like Docker is, so we have a place where we can release and iteratively improve, on what's there. I would say, also because of the pandemic, I think we've been a bit less coordinated than I would like. So we haven't been sort of driving the troops in a particular direction as hard as we have been able to in previous years. But I think we will resume that as we get past this lull period and
sounds
join
awesome,
forces, maybe.
Actually, I don't think that something being a command line tool that should stand in the way of it receiving plenty of attention for what you've accomplished there. That sounds really great. Yeah. So how about the attention thing? Like why so few labs?
Right. I mean, globally, there is a community of about 1,000 C. elegans labs. So start from that set, but then the ones that do neuro, okay, are a subset of maybe, I don't know, 50. Let's say 50 of that set. There's some that are doing very straightforward genetic work of like doing knockouts, seeing what happens to the neurons. Okay, so then you get down to the ones that are using the really advanced techniques. And I think there's again, like some resources stuff, right? So C. elegans are super easy and cheap to like do genetics experiments and knock stuff out. But to do the kind of work that we're talking about here, you need to get a lab that gets enough funding to afford some pretty crazy microscopes, right? To be able to bring the expertise to we're talking about like light sheet microscopes here. And we're talking about being able to put the best calcium sensors in to the cells. There's even some voltage sensors now, but even the best labs have been a little hesitant to use those methods. So there's just, it's hard work on the experimental side, and there's a lot of, I'd say, resource and expertise hurdles to jump over, even when you're already kind of bought in on understanding, like being a worm biologist or physiologist. And so I might be being redundant. You're asking me why is it a rare set of skills? And I'm repeating back to you, because it's a rare set of skills. But I mean,
no, but there's a scientific question in here, because I mean, it sounds like a logistics question or a political question almost, but it is actually a scientific question, because, there has to be a reason why, say, making projects with enough funding to get this done isn't sexy enough. And is it because it's hard to, or can you point out how you transfer what you're learning here from working on the, let's say, emulation of a worm to work with mammals? How you use that? Because I mean, maybe I can't speak for sure about what people say, because I haven't yet interviewed many others, but I'm wondering if one of the things that they say is, well, we have our own line of experiments that are leading us to something useful in mammalian brains, and I don't think we can really use the same tools or the results that are coming out of this C. elegans work. I'm not sure if that makes any sense to say that, but how would you respond to that? Where would you find that connection? It's transferable knowledge.
Yeah. And I'll tell you, in past years, I've hesitated to say, here it is. Once we solve the worm, then we'll move right on to fly, and then we'll move right on to rodent, and then we'll move right on to humans, and it's going to be great, guys. Because I think there is a certain amount of, I don't know, scientific conservatism or soberness to realize how big the task is even at this level. And so when one says, yes, it'll be transferable, one almost presumes that you'll have a satisfactory answer for what's happening at the level of the worm's nervous system in some reasonable amount of time. And that's like predicting when research is going to hit a milestone. But
it does come back to your original reasons for getting into work on C. elegans, right? Because now you'd have to kind of go back up the ladder. You're going to say, so I've done this C. elegans work, and these are the things that I came out with that, and now I'm going to take this, and this is how I would apply that on the next step of the ladder. What would you do? Like you, personally.
Yeah, well, I have some ideas. But they are... again, I think there's some resource challenges, but so I have my own private guesses for some algorithms that currently exist that feel like they have enough of a similarity to what's happening in those 70 to 120 neurons that I think there's enough to play with to start to see if we can go back up the ladder. But when I say it's like my personal pet theory, I kind of haven't written the paper, it's not peer reviewed, and I'm hesitant to kind of declare that this is the direction to go in without some additional kind of backup work, I guess. And it's also a kind of thing where, theory papers are hard to get published in this space and hard to get believed also, because people will be like, well, okay, that's a nice hunch, guys, there, but then what does it even mean? So I find that, even neuroscientists that I talk to are a little bit like, I think you still need to convince me that makes sense. So I think there's a little bit of being trapped in the gaps between different disciplines, right? Where, because I'm cross-trained as a computer guy and a neuro guy, right? When I flip back from neuro to computers, right, there's not a lot of people that have a conversation with me about why what I'm taking from neuro is applicable to the computer side, and like, why do I think so? So, but yeah, basically, and not to keep you in suspense, but I think personally, the area of what's called reservoir computing or echo state networks is, it's not that there's a one-to-one mapping, but there's, I think, enough of an analogy there to start to make some progress back up the chain. But not, I still think it's not, I mean, again, I haven't proven it even to myself enough to, like, there's more that needs to happen in Openworm even to give me more assurance, but that's my current, best guess.
I see kind of a branching of targets or goals here, because what you're saying is you're looking at results from Openworm and potential algorithms that you can then apply to computation, to say artificial cognition, in a sense, something AI related. Right? Yeah. Whereas instead of, say, looking at it and saying, well, here are some things we learned in doing the Openworm project that you can apply to, say, the mouse connectome project or something else that's coming up, right?
Right.
Different direction. Are there any people that you know, or are you involved in planning any sort of milestones for some transition from things coming out of Openworm to other, I guess, large neuroscience tracks?
I'd say I'm doing some informal side research, on the side right now. There's nothing formal at the moment. It's nice to be having this conversation because honestly, there's a lot of other stuff going on in my world right now that's squeezing my time for this kind of down. But I think there could be. Yeah, I think there could be. I think it's a good time for it, actually, given, I think the state of things. So, I mean, I'll give you a sense, right? So, a few years back, I contributed to this article. I think I pointed the other gentleman, I'm sorry, I don't know who it was, who I can look it up, who I spoke with some months back. It was this, it was asking this, it started off with the premise that it was trying to answer the question of, like, how much computing power does the human brain have, right? And it started to unpack the question itself as being problematic because we're not really well grounded in how we make that analysis, right? So if you go to the computer, right, the main performance metric is floating point operations per second. And when you go like, well, what is an operation in terms of the brain? You start to run into these things of like, people have different opinions about
at what level. Yeah,
exactly, right? So and so that means even when you go down to the level of tissue, right, and you're like, aha, here I have a connectome. Oh, here I even have all the cell morphologies, and I have the synapses, and I have the gap junctions, and then if you go beyond that, it's like, oh well, I even have the right dynamics of the ion channels opening and closing, so I can solve the equations that tell me how the neurons change over time. And there's all this complex machinery, right? But fundamentally, we don't know, right, how a bit of tissue is computing. We don't know really like what is the information, the most important thing that's actually flipping bits, if you will, right? And how much of it is just stuff that's swirling around that's not really flipping bits, but is facilitating the flipping of the bits. So what I think is the thing to operationalize, to go back up the chain here, is, again, if you come up with this algorithm at the level of the worm, of what is the fundamental information processing thing, the thing like, Alan Turing's, tape, the tape metaphor, right? And you come up with that, then the hope would be that, okay, sure, you can look at any bit of tissue, right? And there's some differences in the tissue, like the cells a little bit different like this, there's these channels and not those channels. But, the idea that evolution is your guide, nothing in biology makes sense, but in the light of evolution, that you can start to see like, oh, well, in order to have any organism that does anything, it had to have these sorts of dynamics. It was having this basic algorithm, and then everything else is like an articulation, right? Or a further specification on that macro algorithm.
This is really fascinating. This, I think, is the crux, I think, of what can be gained out of the work that Openworm is doing, at least for the neuroscientists, and where we want to go with that. Because this is I've run into exactly the same problem. It's so literally the same problem, you might not even believe it. As in, I was writing a paper for this company working on a new kind of computation called Fathom computing. And the paper was basically, what is the computational power of the brain? And of course, others in the more AI field had tried to get at that, looking at things like counting neurons and synapses and stuff. And then I kind of backtracked and said, well, that's not how you want to compare those things, because it's really about, okay, this is how the brain accomplishes a solution to this problem. This is how an AI would accomplish a solution to this problem. And you get the same result, the same behavior. So how many operations necessary for this cognitive result have been done in the AI versus in the human brain? And that's the chain you need to make between the physiology up to what's actually being accomplished, to give you an idea of what is the computational power of the human brain. So how can it solve problems? So it's always about, you're comparing it with like how the evolution has guided you to be able to solve things. That's the same thing as saying this is how we solve problems. It's how our behavior is being formed, right? And I think that is ultimately what people are looking for when they say, we're scanning a piece of tissue, maybe Drosophila, maybe mouse brain. And now we have this connectome, and we want to get from this to something that behaves like a fruit fly or behaves like a mouse. How do you make that connection going up that from physiology through those equations up to the equations that actually matter at the level where the behavior is happening or where the cognition is happening? And this is something where I guess in Openworm, it's the first case where I really want to look at now your console, your command line program, where you've really made that connection. You've collected data, you've figured out how to take that data and turn it into a model, a functional model, and then how that functional model, how it relates to the behavior of the worm, so that you've made that whole series of connections. And just learning how those things kind of tie together and how you get from what you recorded, the data, to what you really need for your, let's say, worm cognitive model, that's got insight. There must be really important insights in there that you can, in some sense, transfer to work with, Drosophila genalia or mouse at Allen or wherever, right? Or at least I'm hoping that would be the case.
Well, that's the thing, right? So our approach was outside in with Openworm, right? Where we, started from characterizing the motor stuff as the outside and constraining movement to muscle behavior to motor neuron activity. Yeah. Because then one synapse above that is the neurons that you care about. And so most of the simulations are sort of there, but then that's the window that you need, which is like looking up from the motor system to what's sending you signals, right? And why are the patterns to the motor system the ones that they are at the moment that they are? It's because of the output of some information processing thing that's balancing all the things that a worm needs to do. Like, why is it crawling forward now instead of backwards? Why is it going to the left? Why is it going to the right? It's balancing out, like, avoiding predators and mates. And so the pattern of like signals, it's coming down to the motor system, right? Again, that's what these 70 to 120 neurons are doing. So the simulation today is really more about constraining the motor system and like locking it down and saying like, okay, this is how we know what that's doing. But what the next step is this stuff that I'm talking about here, which isn't yet represented in there. But it gives us the window to build from here up into that and constrain it more, and also gives us the opportunity to start bringing in, recordings that have come out of which we've had several different data sets that have come in, but then the work itself of that constraint also takes resources. So anyway, but yes, it has the opportunity to do those things.
Yeah, I think that's really fascinating. It's actually one of this sort of thinking about what actually, when you're looking at the physiology, how do you take out of it the aspects that are really meaningful to the problem that evolution was trying to solve? That's also, I know it sounds weird, but it's one of the reasons why I point people to the neuroprosthetic work that the Berger Lab does. Because, there are a lot of critiques there about, there are way too few electrodes. How could they possibly capture useful stuff and blah, blah, blah. There's a lot going on there. But the really important aspect of that is that they have a system that needs to work with the neural code, and it has to be able to carry out a mapping between CA3 and CA1, and you need to be able to measure behaviorally relevant results, cognitively relevant results. So how has the encoding of memory changed in these patients with dysfunction with this system in there versus when it's not there? Because at that level, at the level of how their results in the experiment change, that's the level of what has been computationally achieved with that neuroprosthetic. Right. And that's where that neuroprosthetic has picked up on something that was in the signals that are in the physiology and has used that in a computationally significant way. Right. And I think that's one of the main things that you can get out of that work. Yeah. So it's all about this translation effort that we're trying to do. Yeah. By the way, Michael, I kind of didn't leave you an opening, and I thought you might have some questions that you had set aside as well. Don't want to eat up the whole time. Are you still on?
Yeah. Yes, I am. Thank you. Let's see. So you said that one of the biggest issues right now is getting enough collaboration between all the different kinds of research areas that are necessary to make this progress and happen. Do you have any ideas about how we might be able to, or by we I mean like if you had like a plan or maybe an idea of how to maybe solve that issue? Do you have maybe some ideas on how to make that happen? Because it seems like if the only way to do that is to get some more results to be able to like prove the legitimacy of the process, then that's kind of like it doesn't really solve any issue, because in order because in order to get those results, you need that collaboration, and so that doesn't really like there isn't really like anything to start with. So like other than having some results to kind of get that motivation and get that collaboration going, is there maybe like some kind of change in communication between the different between the different fields of studies? Maybe I don't know, maybe some kind of a publication or something that can at least bridge that gap between like you did make a lot of references to. There's these gaps between the computer science side and the biology side, and there's so many different kinds of research aspects that nobody wants to take on different multiple focuses, because that's too time consuming, and the expected results of each of those is just so far out that no one really wants to. People just get trapped in their own focus. So yeah, I think, is there maybe something that can be done to maybe develop those interests between the different areas of focus, and maybe get some collaboration going that doesn't involve just getting results to prove that it works?
Yeah, I mean, I think that's the question. Look, I think I believe that there is a faster path and then there is a slower path. And I think the slower path is that over time, you know, as science moves forward, folks do start to see the connections between unrelated fields, and they start to work, you know, together. So like science today in 2021, like a neuroscience lab right now is using, you know, all sorts of like computational science and molecular science and physics and behavioral work and just like all sorts of things. So I do think that like the slower path is that over time, you know, PIs will emerge who are doing more and more stuff that connects this together, and the community is going to learn from itself. And I think over time, right, so these smaller labs are going to eventually fit some things together, and there's going to be some results that catch people's attention, and then there may be more of a interdisciplinary thing that comes together. That's the slow path. You know, this could take a couple decades. I don't know. The faster path is if somebody wants to actually fund it and wants to build an organization that tries to do that faster and put people together, even though they don't necessarily organically build labs around these different technologies, right? And to some extent, we try to do something like that. I would say, being inexperienced organizationally in nonprofits, we haven't done that yet on the Openworm side. I will say another thing that I got out of this decade was learning how to do things in a for-profit context, which seemed to go better. But also the challenge is, you know, like, well, there was a reason why for-profit was really good for bringing hearts and minds together, because, you know, there's the nonprofit good for bringing hearts and minds together, because for-profit also comes with some other disadvantages. But so personally, I'm going through I've been going through a mulling period organizationally of what to do with Openworm and other things. And conversations like this, frankly, are great, because I'm not having them as much as I would like, to like stimulating thought in this area of what can come next. But I think that there may be something more like a public-private partnership that maybe needs to happen in this space if it's going to happen in the short term. Something where, you know, we can avoid conflicts of interest. Like, funding Openworm was always complicated. It's like, oh, well, we'll do something and pharma's going to pay for it. And we're like, but that's really not the ideals here. And what exactly is pharma going to pay for? And I know a lot about why pharma does not pay for stuff like this. It's really far out. Now I know a lot more about that than I did 10 years ago, but also just in the principle of it, it was tricky. So I think there's like how to create the right organization of people and resources to make it happen faster. Otherwise, there's a sense that I have that it's inevitable, I think, that it's going to happen, but just a lot more slowly and a lot more randomly.
That sounds really similar to a lot of the discussions that we're having and a lot of the thoughts that we're we're mulling over and possibilities, and also the different tracks that I've tried, with the for-profit approach and the non-profit approach and the benefits. And it's true, nonprofits, even though nonprofits don't quickly attract big players and money in that, they have the one thing going for them, which is a tenacity, a certain persistence. They just keep going because there's nothing that prevents them from keeping going. And all the people joining in don't need to worry about whether or not they're being taken advantage of, because it's it's a nonprofit organization and open.
Right.
So there's that. So I guess as we're concluding, as we're getting towards the end of this conversation, I maybe it's time to ask you the question that I'm always asked whenever people are interviewing me about whole brain emulation and mind uploading, which is so I'll convert that to your version. Then they always ask me if I want to do that someday. So when worm emulation finally becomes fully available, do you plan to take advantage of it yourself? It's not a serious question, so I'll try to answer it seriously.
Yeah, well, it's funny because I have also gotten involved in some science fiction, thinking about like a future world where there's a much closer connection between biological consciousness and artificial consciousness, and so some of those they really kind of are more like thought experiments than are art, but like thought experiments that have given me the opportunity to think down the road, a ways. And I think the questions that come up for that is like, well, if my consciousness was uploaded to a computer, like, what is that computer connected to, and what can that computer do, and what, am I just trapped in a desktop under the desk? Is that, and somebody can flip me on and talk to me and then shut me off? Right? And that doesn't seem, that's okay. Maybe that's enough. But some of the science fiction thinking is really about like, well, if we got good at this and society's got good at this, then that would drive a market for elaborating all the things that artificial consciousnesses could do in the world. And like, okay, so now it's connected to the internet, but what does that even mean? Like, what are the effectors here? What are the sensors here? Do I get a robotic body that can move around in the world? That'd be pretty nice, right? Is it that kind of science fiction? Or is it the Walt Disney model where I just have to hope that at some point I'll wake up in some sort of a science fiction world, or maybe not, right? Like the brain, like, I'm talking about the cryogenics stuff, right?
Yeah.
So I think it's like, I have to turn the question back around and be like, well, what am I getting here? What am I signing up for on the other end once it's done? But I don't know that's my main motivation for this, is to, live forever, have my consciousness exist forever. I think it's just, for me, it's always been more the pure curiosity of it, and the deeper understanding of oneself. If we can, reach that level of understanding of the brain and the mind, and we've always wondered about the human experience in all sorts of different ways, and artists have tried to capture the physical form to greater degrees of perfection, right? And I think capturing, the human consciousness is a similar kind of achievement of humankind, is to really know ourselves. And I also have another side belief that there's a lot of hidden technological advancements that are unlocked when we understand how our brain processes information in particular. Like, I think it ought to be possible to compute at much lower energy cost, is one of my thoughts. If we really know how the brain is working, I think it ought to be the case that we should be able to radically reduce our power consumption for some kinds of computation. Maybe not every single class of computation, but some kinds of computation should become very energetically inexpensive. And I don't know what else might happen, but like, that would be one that I see more like medium term after we really unlock this. And I don't know, maybe something in there is a way to do something that engages the profit mechanism to then actually fund the research. Because at the end of the day, not having funds coming into this particular part of science, I think, is one of the reasons that it's been not moving very quickly, or as quickly as I would like. I'm impatient, so maybe it's moving quickly relative to some metrics.
If you look at the whole span of history, it's all moving very quickly right now.
That's right.
But, yeah. For us personally, it always seems too slow.
That's right.
But those are some really good motivations. And I think that's a great place to end the interview as well.
Okay, many thanks to Dr. Stephen Larson for that interview. Next up, we have our interview with Dr. Natasha Vita-More.
Dr. Natasha Vita-More on C. elegans. Dr. Natasha Vita-More is the Executive Director of Humanity Plus Incorporated. Dr. Vita-More is the author and co-editor of The Transhumanist Reader, a project innovator for future body design, Primo Posthuman, lead scientific researcher of the Memory Project, and contributor to the project conception of the regenerative generation. In the following interview, conducted by Dr. Randall Kuna, Dr. Natasha Vita-More presents her research interest and experience with C. elegans in the context of cryopreservation and revival research.
Hey, Natasha. Thank you very much for joining us. And as you know, I'm going to ask about your work in C. elegans, but if you'd like to, maybe it'd be useful to kind of situate your work in your overall endeavor and your goals, and what led you to then decide, doing a study on C. elegans, that really fits into what I want to do. Where does that fit in?
It's interesting because when I originally started getting interested in longevity and memory and cognitive properties and consciousness, and why not back up the brain and create new bodies, I developed a future whole body prototype based on nanomedicine and artificial intelligence and automation to a degree. And through that, the concept of the metabrain emerged, which would be a third brain. The term was coined by Dr. Greg Fay and Dr. Max Moore differently, and I came up with the idea probably because I read Fay's and Moore's work, and I thought it was a perfect term to talk about this third brain, this tertiary brain that we could upload our memories into and download them back into this whole body prosthetic. And so that led me to start thinking about the vulnerability of the brain itself, outside the vulnerability of biology, and the need for backing up or storing memory. Simultaneously, my mother got dementia, and we took care of her for the last, 4 years of her life here at our home in Scottsdale and hospice. And she lived to her 100th year, but with dementia. And she was such an incredibly intelligent and straightforward person that had enormous potential, especially during her era, that it really drew me to start thinking about what are the alternatives to aging and death, and what are the alternatives to the neurological deficiencies. And so that led me to start thinking about work that had been done in the scientific arena outside the design world, and addressing the issue of long-term and short-term memory.
Long-term and short-term memory. Oh, wow, I have a lot of questions right now just because of what you just told me. First of all, maybe for anyone listening, it might be useful for them to know what are the first and second brain if the third brain is the meta brain. Okay,
so the frontal lobe grew out of the whole brain from the fight or flight reptilian of our brain stem to the cerebrium, to the cerebellum, and looking at the frontal cortex. And the ideas have always been that, do we use all of our intelligence? Do we use just 10% of it? What percentage? And that's never been proven or disproven. But the idea that maybe artificial intelligence could help us be better at solving problems, being more humane individuals, more empathic, more aware of our actions and our reactions, our ability to address feedback, the cybernetic system of the brain, feedback and control, the whole Rashomon principle, and taking a look at, do we really remember what actually occurred, and what would it be like if we could have instant replay of any moment or minutes of our lives to assess our reaction. And it's almost like the gamification of cognitive properties. And so the meta brain was the idea that with artificial intelligence and nanomedicine, that we would be able to store our memories outside the body for the purposes of, as you wrote, whole brain emulation, the concept of uploading, downloading, but I see it also as cross-loading onto multiple substrates, depending on which ones are the healthiest to exist within.
Very nice. So in the context of what you just said before about studying, I guess, what was working memory and long-term memory, is that what you said, or was it short-term?
I said short-term, but basically working memory, yes.
Yeah. And how does that apply in an animal like C. elegans? I mean, do we even know that C. elegans has both of those?
Yeah, that's really interesting concept. Yes, those neurons have been identified, the ones that retain long-term memory, which isn't very long for C. elegans since their lifespan is, 2 weeks. Mm-hmm. And then short-term memory, just working memory. They're interrelated. My research was in long-term memory with C. elegans. And I know that you're probably asking what led me to the C. elegans. Why did I choose this simple animal? Yeah. Okay, well, I'd love to explain that as briefly as possible. When I thought about doing scientific research, it largely stemmed from the field of bioarts. I'd been in the design world and the performative arts and literature, and I thought that there's so many different designers and hackers, biohacking organisms, and some astounding work by Stellock and Martha Menendez and Laura Beloff and numerous doctors in multimedia design and science that were doing incredible work with experimenting with genes and transgenesis, et cetera. And I thought, well, that's one way I could do it. Maybe I could do a scientific experiment, even though I had not done one in the past. So I searched and researched the most highly respected, I guess, organism possible. And the candidates were few and far between. There was the C. elegans and the fruit fly. I chose a C. elegans. Its functions applied to, in the research, applied to aging, like the fruit fly. But there have been research on the neurons of the C. elegans, the whole 102 approximate neurons that function. And the beauty there is that they are transparent. You can see the neurons as they're working throughout the animal. It doesn't have a brain, so they're aligned and functioning throughout the whole body system. The connectome of the C. elegans. And specifically, the C. elegans was the first organism to have its full genome sequenced. And that was really impressive to me. And that was done in 1998. And I thought, well, here's the first multicellular organism to have its whole genome sequenced. And how cool is that? And further, also the nematode, this C. elegans, it's a tiny worm, microscopic, has a single gene mutations that have been researched and identified to extend their full growth lifespan. And that rolled out in Nature magazine around, I think it was 2000, and that was when I first started thinking about doing this research. So it built up slowly. And then, of course, my advisors, Greg Fay and Ramon Risco, both very skilled in gerontology and evolutionary biology, they influenced me to go towards the C. elegans. But I just want to say that I had already known about Dr. Judith Campisi and Dr. Cynthia Kenyon, and of course, Dr. Michael Rose. Judith Campisi had worked as a biochemist and cell biologist, had worked with the nematode C. elegans and identified a gene which had its telomerase enzyme shortening. So they were able to see that and identify it, which was really phenomenal. And also Cynthia Kenyon's work with C. elegans is very well known in proteins and the proteins that affect aging. And she identified the DAF-2 gene mutation. So I thought those issues were really important. And because I needed to have primary sources and to identify prior research that I could build my work off of, those Cynthia Kenyon and Judith Campisi were very influential, along with Greg Fay and Ramon Risco in their fields, and that influenced me in that direction.
And so what was the goal of your study? What did you hope to get out of it? What was the setup and how did it go?
Yeah, the endpoint goal, whether I succeeded or not, was to try. And, one foot in front of the other. And it took a number of years to actually develop my protocol, my methodology for this research. Going in, as a novice, as someone who hadn't worked in a lab, I trained at 21st Century Medicine. I learned how to work under the hood, which is not very easy, how to pipette with great difficulty. I kept on pushing the wrong way on the pipetter. How to keep your Bay Area clean. Just the protocols of being a scientific researcher in a lab are very important, and also learned how to look at the C. elegans, how to study it and understand its functionality, its goals, identify that there's a male and hermaphrodite. There's no females. The hermaphrodite is the one that reproduces, but a male can reproduce as well. So learning the behavior of this animal was really exciting to me. So my goal was to test memory. And that goal had already been identified by me earlier in my doctorate in life expansion, thinking about memory. As I explained earlier, my concern with it, with the meta brain and what would happen in the future with AI and being able to back up the brain, and noticing the advances in early onset Alzheimer's and dementia among humans. So that worried me a lot. And I thought, what can I do? So I didn't know if it was going to be long-term memory or short-term memory or working memory that I was going to focus on in the beginning of my research. I just knew it. I had to work on memory. And with choosing the C. elegans, I narrowed the protocols that I would apply. I decided to use vitrification, which is a cryonics process with quick low temperature freezing that's used for freezing embryos and sperm and eggs. Highly regarded, so I followed part of that protocol. I also knew that C. elegans had been successfully suspended in cryonics or cryobiology and survived. And I also knew that C. elegans are known to be good learners. You can teach them a task and they will learn the task. And that the tasks are best learned in C. elegans through sensory makeup, sight, light, where the light is shown in the Petri dish. They'll either turn away from it or go to it, sound tapping on the dish or making noise. They'll either adjust to or retract from, and smell. Their olfactory sense, the ability to smell, is their most profound sense. And I also was well aware that in humans, our smell is largely associated with our memory. So that was, yes, check that box off. Olfactory is what I had to do. I had to train them to do something through smell. Kind of Pavlovian, but not too Pavlovian, because it had to be, a solid scientific research that I could repeat. So the end goal was to test them, to train them to do a task, to vitrify them and also put them in cryonics, two different protocols there for suspension in cryobiology, and then to wake them up, to revive them, and to test them to see if they remembered the task that I'd taught them. Well, the issue then became, what is my goal for the task? And the easiest task for any of us is food. So then I determined, when they were little babies, just little larvae, after they hatch out of their larva state, to place a certain smell, a chemical in their food, and watch them in the petri dish go to that food and have them learn to associate the smell of that chemical with food, feeding time. And so the goal was then to test their memory, and it turned out to be long-term memory because I could only vitrify and preserve them at an adult stage because they had to practice that enough to become adults. And then before they got too old, put them in cryonics and then bring them back to life and test them. So it was a very finite window of opportunity there. And I also had to work with hundreds of these worms, these nematodes. So in short, the goal was to test memory and to see if memory, the neurons could be vitrified or cryonically suspended, let's just say frozen to simplify, and if they could be woken up and if they would have their integrity afterwards.
So if they still remembered the long-term memory, you trained them once they came back from suspension?
Yes.
Yeah. How many of the worms, do all the worms survive cryonic suspension by vitrification?
Gosh, that's such a delicate question. I worked with hundreds, or we worked, my team worked with hundreds of worms, and ordered them from Michigan University, which has their lab there, and ordered my chemicals there. And I had to learn how to do all of this and how to, take them once they arrived in their package and put them in, the petri dishes, and put them in a really healthy environment that was first and foremost. Considering that they are simple animals, I wanted them to be as healthy and their environment to be as safe and comforting as possible. Learning how to do that, I would say we worked with hundreds and hundreds of worms. Hundreds died, hundreds survived. But in our final testing for the evidence of the scientific breakthrough, it was hundreds. 200 to 300 worms that we used as the final assay in sampling.
So was it just a matter of figuring out the right protocol, and then when you have that, then most of them survive, basically?
Yes.
And so I know this isn't really directly about the connectome and about all that, but I'm just very curious because I haven't worked with C. elegans at all myself, so I don't have any basis of understanding really. And I'm just trying to wrap my mind around why C. elegans seems to be so good at doing that. Why can you vitrify C. elegans and then bring them back and they seem to be fine? And it's so much harder with pretty much most other animals that you could try, and certainly very, complex brains. Do you have any insight into that?
Yes, I do. I think first, while they're microscopic, they're not so small that you can't see them. You can see them easily with a microscope. And I had to purchase numerous microscopes to find the correct one that I could use and also put a video camera attachment to. But they're easy to see. Not only that, they're beautiful to watch. They are lovely dancers. They move through the agar in the petri dish in this beautiful fashion that is just, they're wonderful to watch. Also, they have their neurons, their 302 approximate neurons are throughout their entire body, so you can see their neurons. And that's quite wonderful. I mean, with your naked eye in the microscope, you can watch their neurons. You have to, balance it, just right. But watching them in the petri dish is a beautiful thing to do. They have an incredible sensory system. As I said before, they adapt to readily or reject readily sound, light, and smell. So that's really good to have an intact sensory system that you can identify and work with. Also, they have a strong toxicity resistance. I think on the same level as rats, they don't react so much to toxicity as other animals, humans do. We are we get hives and all sorts of reactions. They are pretty stable in that regard, and their lifespan is short. It's only a couple of weeks, so you have to work rapidly. And I think there's some benefits to working rapidly. You have to be on your best game, 24/7 and working with them and have it really timed down to, a really fast-paced and detailed production. So you can move through it and experiment. And if one assay doesn't work out successfully, and you decide to change the protocol, you can do it more quickly than if you had an animal that was more complex that you had to spend more time with, perhaps.
So maybe that is a prime reason why it works so well with C. elegans. It's just
I think so.
Quickly, there isn't the time gradient there.
You can see them and they're beautiful, and they're not like fruit flies or bats or there's some sea life that chronicists want to work with and test them, and some of them are cute and some not so cute. So I think it's almost like, I don't know, there's this familiarity with the C. elegans because they move so beautifully and they're in sync, and it's just, I'm sure I could, espouse far more credible reasons, but I think mine was more sensorial and based on the fact that they have been known in the scientific field as the model, the elegant model research organism to use, especially in longevity.
And just the fact that you could bring them back from a vitrified state and they work, and therefore their little brain, it's not a real brain, but throughout their body, their neural system has to be intact to work. That gives a lot of confidence that when you prepare them and look at them under a microscope by making a bunch of sections, that you're actually looking at something that is a connectome that is intact enough that it would be representing something that you could then try to make a functional model for if you're trying to do whole brain emulation.
Yes.
So that's the interesting part about that. You have this extra assurance kind of that it's the real thing. So that I see the real benefit of working with this tiny animal, especially that you can also simultaneously see the neurons and you can do functional recording that you can match up with the connectomic work. So I was kind of surprised that there is actually quite a lot of pushback against working with C. elegans when you ask many neuroscientists about, well, why aren't you working with C. elegans? Why did you pick something much more complex like Drosophila, even though, it's not the most complex animal, but it's way more complex than C. elegans and or mouse, or then straight to humans. And some of the reasons I got when I asked is, well, the neurons typically don't seem to spike. They seem to be sub-threshold, so it's a different kind of domain that they're working in, and so maybe it's not a great model to learn from. But even then, it feels like you could learn a lot about the methodology, the procedure for how do you go from a connectome to something functional. And so I wasn't entirely sure if it made sense to just reject working with C. elegans for that reason. So maybe there's another reason why it didn't get as much attention as it should. Do you have any idea where that comes from?
I think, let's take a look at it. A fruit fly, however small, here in Scottsdale, we have no-see-ums. They're so tiny you can't see them, but they bite you. They're like vampires. They have very sharp teeth and they draw blood. They're like gnats. You can see them. They're a bit annoying, but they move very quickly. And when they move very quickly, they're out of touch. You can't, grasp onto them. So that would make the fruit fly not so attractive to me as the C. elegans, which you can watch in the Petri dish, and it's within a confined area, being the Petri dish, no matter the size of the Petri dish. As far as the rat, I know that Roy Walford, of course, worked with mice and caloric restriction, and rats have been used a lot. And rats have a higher level of intelligence. They have cognitive properties that C. elegans do not have, or fruit flies do not have. They're not constantly moving and flying around where you can barely see them, so they're easier to hold on to. However, I think the beauty of the C. elegans is the connectome. And I think also, going back to looking at our prior conversation, their sensorial makeup, the fact that they do react to certain senses, and they're willing candidates. They're not going to fight you like a rat. That's going to make you say, oh, I'll go lose it, go away. I'm more interested in this over here. They are more cooperative, perhaps. They don't function so much in schools, although they do flow in a pattern. They are different layers in the agar, in the substance in the Petri dish. So you have to change your microscope to see the different layers of how they move. like schools of fish. But I guess part of the pushback would be that they don't have a brain. And that could be part of it. So maybe they're not revered as a higher level intelligence, but they are still a simple animal, and the neurons still function, and they can be taught. So they learn tasks, and they can remember the tasks that they've been taught or they've learned. So I think that there is a viability and integrity there in this simple organism that gives it the credibility that may be dismissed by others who would find working with a brain per se offers more validity.
Yeah, I know that, of course, the brain is very different from a human brain, and they don't have a brain as such. But it seems to me that it's exactly the type of things that we do have in common with them, where actually, for instance, connectomics in mammals is struggling. So when you look at an image stack collected from mouse or from any other mammal, one of the big problems is to try to identify the type of gap junctions, because gap junctions, the electrical junctions that can have transmission properties that are more like the transmission properties between the neurons in C. elegans, where it's subthreshold. Those are really hard to distinguish looking under an electron microscope. And so it's precisely that part of the problem that you have also in this 302 neuron animal, where you could learn a lot about how to identify the type of synapse that you're looking at in a much simpler model, where you can look at the activity at the same time. So it just seems like a useful thing for learning methodology, no matter what. So yeah, anyway, that's just my rant, I guess, about why not go there and why yes go there. But anyway, enough of me. Where do you think that studies should go next? What do you think is kind of the most next promising thing? And broadly, it doesn't have to be about C. elegans, it doesn't have to be more of that study, but knowing what you know, where do you think we should go next?
Definitely backing up neuron function and the electrical charges between neurons. I think, on the human, that's where we need to go next. And I think my research, did set a precedent. It did accomplish a scientific breakthrough, however small and tiny. It succeeded in what I set out to do. I reached my goal. Rolled up my sleeves, got my hands dirty, and then thanks goodness to my lab technician, who is far better than me at pulling them into the straws. I mean, if you take a look at the research, it's pretty amazing how we had to get them into the Petri dish and into the liquid nitrogen and move them around in a straw.
Did they walk freely into the microscope nitrogen?
Trying to pull them into the straw to see what we were doing. And that is an invention of Dr. Ramon Risco in Spain, who is a cryobiologist. The next big step outside of anything I've done, and I think that where we need to go is looking at the neurons, the electrical charges, how to back them up, how to copy them, to find that information, and that leads us to your work. And I think that the rest of us can come up with scenarios, theories, possible futures, all sorts of ideas that you in your field can build out. One area that interests me, which builds off of the concept of that originated in 1996 with the whole body prosthetic with the metabrain, is Elon Musk's Neuralink. And I think Neuralink, he does mention it's a tertiary, a third brain, and it's while it may not look the same, I think the end goal is very similar: to restore memories, to back up memories, to copy them, transfer them, restore them, identify them, and see what those connections are. And I think that this is the next frontier for the human, not only with our memories and our identity, which can have a personal identity or who you are without your memories, so that continuity and that seamlessness. It doesn't have to be so seamless. Sometimes you forget what you did the day before, but at least that ability to find that information and to have it stored is so on point with where we are today, especially in advances in technology and the use of microchips and computerization everywhere and advances in AI, it would be foolish and foolhardy to even dismiss this at this point, that we have to back up our brains. We have to understand it and do it, even if we don't know what we're doing. As Anders Sandberg at the Future of Humanity Institute, Oxford University, has always said, as a neuroscientist, we don't have to know the roadmap exactly. We just know that we have to go in that direction. And I agree with it 100%. We'll learn along the way, much as I did in my research. I went in very green, and I learned, I did the schooling, did the practice and developed, and then found my way to where I did succeed. And it's frustrating, and it can be hard, and you scream in your pillow at night because, oh, it's so frustrating, but you stick with it and you succeed, and that's what separates the doers from the non-doers. And it doesn't have to be always correct, it doesn't have to be perfect, and you don't have to always succeed. It's the ability to try, just to do it, that makes a difference.
So I think right?
Yeah, I think that Neuralink is one project. I'd like to see more of these projects. I think you've been in this area for so long that finally the world is catching up to you, Randall. So that's quite wonderful to see.
Give me too much credit.
No, it's true. I mean, we know this, and it's about time. And sometimes we have to sit back and wait for time to catch up with us. And I've been there. It's hard, but the persistence pays off in the end.
Yeah. And do you think there's something really surprising that you learned in your work? And, mostly the C. elegans work I'm asking about right now, but or something adjacent to it. Wondering if there's something really surprising.
Yes. Surprising, I suppose a couple of things. One, I'll say one of the most aha moments, the beautiful science moments to me was I was working in the lab, and I was under the microscope for hours, and I was looking at a batch that we just brought back to life after the warming bath, after the vitrification. And I was looking, and I said to my lab technician and my cohort on this project, I said, I made a mistake. I blew too many bubbles, oxygen into the dish when I brought the worms, put them into the Petri dish. I said, oh my goodness, I can't tell now what's going on. I was looking and looking. And he said, no, that's fine. That's larva. And I looked again, and I had to adjust the microscope, and I saw, and I have this on film, and it's so beautiful. Mm-hmm. I trained and vitrified a pregnant hermaphrodite. It gave birth after being in cryonics, brought back to revived in the Petri dish, gave birth, laid the eggs. I was watching those bubbles. They weren't bubbles. They were eggs, larvae, wow. And I watched them hatch. And I saw 3 different eggs hatch, and little baby nematode worms wiggle out and go swimming in the Arga beautifully. And it was one of those moments I started crying, because the whole reason I got into life extension, back in 1980, was because I was in Japan working, and I was pregnant, and I lost my pregnancy. And it was just so beautiful. I thought our bodies are so vulnerable, and it really affected my brain. I had vertigo after that, and just really horrible cognitive, neurological shortings because of the blood transfusion and the emergency. I was literally dying, and thankfully I survived that, but I lost my pregnancy due to that. And so that caused me to get interested in the vulnerability of the human body, and especially in reproduction and neurological functions and whatnot, going through the biomedical processes, and then to experience this beautiful little simple animal giving birth after being put in cryonics frozen, let's say for the audience sake. It was a lift. So that was my aha moment. I have pictures of it. I have it on video. So that's wonderful. But Randall, to answer your question as directly as possible, what I learned is the incredible rigor of science and the efficacy of scientific research and the need to write your paper and have it published. And going through the writing part was even more difficult than doing the research. And developing the methodology. It's putting it in that language, the terminology that is on par with the standard for a scientific research. Was really hard, and had blind peer review, and sometimes I would just get a little bit edgy with it because I was doing my best, but it wasn't good enough. So I had to really refine it to where it was finally published. And it is, of course, located at online and published in Aubrey de Grey's journal, as well as on the government's website for science. But
and peer review tends to make ultimately the result is better, right, after peer review, and you've had to go through all those cycles.
Yes.
Don't you find?
I mean, I've have so many different skills that I've learned over the years, because I need those skills to do my research. So I just jump in and learn it and wade through it. This one was really hard because it was coming up with the idea, then really taking it seriously, getting a grant to produce it, then building my scientific team who were more skilled than me to advise me and work with me in the lab, to actually make sure that I did the work myself. I wasn't just hiring someone to do the work. I had to go in the lab and roll up my sleeves and do it. And to watch the beauty of, the survival, the revival of the simple animal. And then to go, wow, and then create the essays and prove it and then write it up. It was a success moment that we don't always have in our lives. You try some things, sometimes they work, sometimes they don't. So that made me feel really good. I never really promoted it. I never, today everyone's promoting themselves all over the place, and sometimes it really gets a little bit tiresome. It's like, I just didn't want to hold up a worm, with my face and Instagram it all over the place. I do a little social media. I didn't have a marketing person. I didn't self-promote it. I thought the work will stand on its own. It doesn't need me to brag about it or, create a company, become CEO of it, of the worm and memory. But I hope that it is used in some way on some other level. And so that's why I'm so delighted to be talking to you. I think that
it's still one of a kind research, isn't it? I think it's been replicated.
Well, it did a breakthrough in cryonics. It proves that a simple animal can come back to life and retain its long-term memory. I think that it's also is outside of it. And again, that's why I'm delighted to be talking to you and to look at your work and to look at the larger issue of the connectome and what does this mean and what is the relationship between these neurons. And then within the neuron, we'll get into some really delicate stuff, and that's where you're an expert and I am not. But I understand from the abstract, the larger picture, the gestalt of it, the importance of that connection.
If you have the chance to join the panel as well on the 20th, then of course you can also have a back and forth with the other people that are there.
I'd love to. Oh, that would be great.
So this is actually, for me, this is a fantastic point to leave it at, because I think that your last description of your experience was beautiful. But I do want to, give both of you, Michael, who's been doing our wonderful technical work here right now, a chance to ask if he has any questions or if he would like to ask you anything. And of course, if you believe that we've left something out, if there's something that you think should really be added that we didn't.
Yeah, in one of your questions, you had asked knowledge about some of the best work in C. elegans connectomics. And I wanted to mention that the research with the nervous system theories that are developed about the neuronal connections, but I think the Deep Worm project is one that you can visualize on a 3D level. Some of the work that has been done in the past has been done for years based on the sequencing of the full genome of the worm and its neuronal mix amongst those 302 neurons. I think that is a beautiful project to visualize. And I think that visualizing it through the gamification of the connectome, the 3D imaging, and helping people understand the electrical charges and the deep insight into digging even further into that mine of wonderment.
And that's the Deep Worm project, right?
Yeah.
Yeah. Mm-hmm. Fantastic. Michael, did you have any questions?
Thank you, Natasha. Yes, I did have 2 questions. So my first one is, considering the successful revival of the C. elegans after being suspended, to start moving towards more complex species, where I think the most obvious difference is like the change in volume, but then there is also the complexity of the connectomics. Is there something specific that is different between the success for reviving a C. elegans compared with something more complex? Like, is there something that still has to be understood about how to go about the process, or is it just complexity? Like, is it just simply that, and it's a little bit more delicate? So I guess to rephrase the question, to start working towards experimenting with reviving more complex species, is it a scientific research issue where there's still questions that need to be answered, or is it more of an engineering issue where the process just has to be revived and made more delicate?
Probably more of an engineering issue. If you take a look at, say, reviving a dog or a human, far more complex in the brain, but not just the brain. It's the central nervous system, the brain and the spine and the peripheral nervous system, and all the information that's going on. Nanomedicine would be truly required for that. Nanomedicine is not at that level, but if you're just a neural patient or a candidate, let's say, and you become a neural patient, and your brain is vitrified, looking at the CT scans of it to see the integrity of the brain afterwards has been shown that those scans evidence there's not the worry of, or so much worry about the freezing of tissue of the cellular structure, which would call fracturing at that frozen state, that the tissue in the brain looks good, looks healthy, it looks like it could be revived. But that's a long way off. I think that if we were to looking at maybe a rat might be a next good bet. But again, the engineering aspect of reviving a body is pretty complex. I mean, just reviving some neurons within a simple animal was complex to begin with. The engineering not as sophisticated as it would be for a larger animal. But I do think it's probably the engineering, and it's clear how to vitrify and cryosuspend. It's clear about the cryoprotectants. It's clear about the process, the tools that are used, the dewar, the liquid nitrogen. All of that has a level of again, integrity and viability that assures the scientific community, even the harsh critics, that there is something substantial there, and we can evidence that through embryo freezing and egg and sperm freezing. So I think embryo is the best example to refer to there. The infertility industry, how it's grown by leaps and bounds because of cryobiology. We're looking at organs now, 21st century medicine especially, but if we freeze organs, all the people that need transplants or implants to be able to have organs would be great. And one way is to have your own organ cloned, put it into cryonics, and then if you need that organ, whether it's a lung or a liver, a kidney, a heart, you have your own, so there'd be a good match and less adverse reaction to toxicity and those issues. So I think that's it's a slow step in that direction. I probably didn't answer your question as directly as you would have liked, but I had to take a roundabout to get substantive.
Yeah, thank you. And I think it answered it pretty well, and it's actually, I think, pretty encouraging because, especially with Openworm emulation, when something goes from a science issue to an engineering issue, that's always a huge milestone. And then my second question was, earlier in the conversation, you mentioned a -2 gene that affects aging. That was also found in the C. elegans, and I was wondering if you could maybe explain a little bit more about what that is, and is that something that is research on that something that can be applied to aging in humans?
I would think so. I mean, that's certainly Dr. Cynthia Kenyon's research. And as a molecular biologist, she is highly skilled in that area. She's been working in this field for a long time. So I can't give you the scientific language that Randall could or Cynthia could in that regard. But while we may not share the same genes and the terminology or the labeling of the gene, as this with DAF-2 gene, there would be something synonymous within the human gene, and I could probably Google that and find out what is the equivalent to a DAF-2 gene in a C. elegans to a human on the same scale. The interesting thing with that research is that seeing that this gene and identifying that this gene, mutations in it are doubling cause mutations in the gene cause the lifespan to double. And that is the significant thing. So and I think Randall's Googling it right now. So if we could find out what is synonymous with the DAF2 gene in the C. elegans to the human, and see if Dr. Kenyon's research could be duplicated within the human, and if there's any type of protocol that is used in that way, that would be really interesting. And I'm sure I'm not the first to say that. So Randall, did you come up with anything significant?
Oh, not that fast, no. I'd have to really look through the Google results a bit more, but I'm very interested in it because, we all want to find something that helps us hang in there until wind uploading as possible, right? So
yes,
we need to find the bridge. Yeah.
Definitely, how to stay alive long enough. And if you do have to go into chronics, to at least know that some of your neurons for long-term memory will probably work is a good thing. And in the interim, we certainly need to be looking at the ways and means to engineer backing up the brain. It's so consequential now, and more than ever. And I think that the last couple of years has shown us this, if not through the accelerating advances in technology, the exponential advances in science and technology, but the rate of change across the platform on a transdisciplinary level, and the desire for people to live longer and to care for those with dementia. And that if we're going to live longer, we certainly need to have our memories intact or as good as possible, and to have our cognizance, our consciousness aware.
Yeah. Cognitive health with long lifespan. That's a great goal. Yeah. Thank you so much. This was a fantastic interview, and I think it actually fits extremely well into our topic. And so I really hope that you're going to be there on the 20th, and you can join the conversation with the rest of our guests.
I would love to. Thank you both.
Okay, many thanks to Dr. Natasha Wiedemar for that interview. Next up, we'll start our live discussion panel. But before we begin, I'm just going to read a few lines regarding how we sort of expect the discussion to proceed. First of all, our workshop is fully participatory and is open to every one of our international attendees and viewers. One way to participate is to write questions directly into the chat that is enabled for our YouTube live stream. From there, one of our excellent staff members is monitoring the chat and will alert the panel to interesting and topical questions. Questions that are more complicated or multi-part, where it is important to establish that the question is both properly understood and answered, are best asked by directly engaging with the panelists. Of course, we make this possible through our Callin Number. The number and PIN are shown at the bottom of the screen. They're also written in the live stream description and on the livestream.carboncopies.org webpage. They are as follows: 786-540-5631 with PIN 180-618-986#. And if you're calling from outside the US, it may be better to use the web meeting option at call.carboncopies.org to avoid any telephone charges. And this entire process is similar to calling into a radio show. Your call will be monitored and a respectful exchange is, of course, expected. When your question would benefit from direct interaction, I can invite you to call in if you can. If you want to add a note to your question, letting us know in advance whether you can call in or not, that can be very helpful. With that all said, does anyone have any pressing topics and or comments they want to start the discussion with?
Hi, so thank you, Bill, and thank you very much to both of our speakers, Dr. Stephen Larson and Dr. Natasha Vida-Moore. They were quite in-depth, those conversations, and I bet we could have gone on forever, but I guess this is a great point to start talking. Basically, I have a set of questions myself, of course, but I would love to let others go ahead and ask their questions first, especially anyone who is here who wants to immediately chime up and just ask their question directly. Maybe we haven't collected it yet in the document where we're trying to collect our questions. I would love to give the floor right now to anyone who would just love to, you can either raise your hand and then I'll call upon you. We have this raise hand feature here, or since there are not too many of us, you could even just chime in right away. I'm sure it wouldn't be too much of a problem. No questions at all. That is surprising, but we can work with that. Maybe it's because they're already back here. Oh, Stephen's not on. Okay, that's, yeah, probably best to start with Natasha then. I wonder, Michael, if you can just ping him and see if he has had trouble finding the URL. That'd be very useful if he could manage to join, of course. Yeah. So let me just ask a question that I was actually going to ask to both of our speakers first, but I might as well just ask you first, Natasha. I was really wondering, given how much further along work on the nematode is than on all the more sophisticated animals, and then, for instance, the Stephen sort of dangled the prospect that we have the first emulation of a nematode in this command line system that they've got, where they've put together the best of what they have. And meanwhile, you've shown that you can revive the nematode, and we have plenty of work, therefore, on both the preservation and on at least beginning the emulation of it. I was wondering, what do you think would happen if someone published and publicized a so-called whole brain emulation of a nematode at this point with this kind of result? Or maybe it would be even more sensational to call it the upload procedure for a nematode. In your opinion, would that sort of thing, do you think strategically that would accelerate work, or would it cause a backlash? And why?
I think it would accelerate work. I loved your description. First, any simulation, whether it's 3D or gamification of a concept, is, when done aesthetically, is attractive, and it's something we want to look at, as long as it's concretized to a short span that people won't be bored by, but can see and get within the first 5 seconds, like giving a pitch. You want to make sure that you get it down within the first 5 seconds. You got 20 seconds to get those investors or that audience to actually go, oh, that's interesting. So I would say that having the simulation would be great. I think either using the upload terminology or whole brain emulation terminology would be great, or to come up with a new way that maybe, if upload does trigger some science fiction crap, and concern that people just want to upload and be located in cyberspace as zeros and ones. You want to make sure that you steer away from that particular misinterpretation and misjudgment of what is going on with uploading or whole brain emulation. And I mean, don't let them read more of that.
Right,
first.
right.
Although I love Hans's work and pigs in cyberspace and all of that, but I think uploading does deserve to be credited in academics and in scholarship as a viable process, whether you use upload or whole brain emulation. But I think that it would certainly draw attention to the possibility of it.
Mm-hmm. And that's despite the fact that I guess nematode consciousness might be
questionable.
So
I mean, and then we say, what is consciousness? And I remember being at the consciousness conference at the University of Arizona, and you were there. You held a workshop, I believe. And,
that's right, yeah.
If you have these heavy duty consciousness seekers who have been in that particular field for a long time, they may bring up some worthy questions to pay attention to. But bottom line is, no one knows what consciousness is. And we can pontificate on it and create conjecture on it. And we still don't know. So who's to say that the consciousness of a nematode, because it doesn't have a brain or a frontal lobe or whatever, it doesn't have consciousness. I don't think that is an issue, really, in light of the fact that no one really knows what consciousness is. That's a different, though, than self-actualization or awareness, self-awareness. Like my puppy on the floor is very self-aware that he's making noise to distract me because he wants my attention. And, 100 years ago, we didn't consider dogs not we, but humanity did not consider dogs largely to be have consciousness. We know that's a fallacy. So who is to say that the worm is not self-aware? Well, it seems like it would be self-aware, or else it wouldn't have long-term memory.
I guess we could probably debate worm consciousness for quite a while, but
I want to Google it, but I don't want to be distracted from talking to you. Does anyone in the audience know anything about worm consciousness?
Actually, more. I'm really interested just in this sort of strategic thing. I know we're kind of coming at this backwards, because usually when we do these workshops, first we talk about the science questions, things that came up after listening to the speakers, and then we start talking about strategy and stuff like that. And I'm going at it the wrong way around by immediately asking strategy question. It's just that it was so like this thing that just popped into my mind. Like, if we have a model like that there, why isn't anyone making a bit more noise about it? And it would be great to hear Stephen's remarks on that if he manages to join us. But yeah, I see Joe has raised his hands. I'd love to hear from other people about their opinions about this. Yeah.
Well,
sorry. Yeah, go ahead.
Shall I? Okay. From what I know of the Openworm project, it's a model that sort of represents a generic nematode, not an individual nematode. And Natasha, when I was listening to your description of your experiment with the freezing of the worms, I was struck by how you operationalized your experiment by actually training the worms to exhibit some behavior and then verifying that they remembered that training after they were revived. And I wonder if we could do the same thing to show some operational measure of success with emulating a nematode by first training the worms to do some behavior and then scanning them in and configuring an emulation and showing that the simulated worms exhibit the same behaviors.
Oh, that's interesting. So, for example, would you say then take my research with the olfactory sense and the interrelationship of those particular neurons that work directly with memory and smell and simulate them? Or were you talking about do a task that would involve self-awareness and potential?
Well, I was dodging the self-awareness function. Which I think for scientific study is probably a good idea.
So what task? I mean, is there any task that you can think of right off the top that would identify self-awareness? I mean,
Right. I don't know. I don't
bumping
know
into
how to
something and having a
operationalize
reaction
that.
would identify. Randall, don't you think that if, like,
Well,
going one direction and it's not safe, and turning around and going another direction where there's happier worms,
yeah,
I mean,
but
that would.
I mean, it's you know, I think the first problem always when talking about consciousness and self-awareness is to get everybody to agree on the definition of what they're talking about.
Yeah.
And that very much depends on whether you're talking to a physician who wants to know, is the patient conscious or unconscious? Or whether you're talking to a philosopher who wants to talk about consciousness from that perspective, and so forth and so forth. And so, you know, if we look at a behaving worm or even a robot, and we say, hey, the robot bumped into a wall and turned around, does this mean the robot is conscious? Is it self-aware, maybe? Even if, you know, those two words don't necessarily mean the same thing either.
It would be self-aware, yeah.
Yeah, so this is or aware of its surroundings, at least, even if not self-aware. It may have no self-model, but be aware of its surroundings and know what to do if it bumps into a wall. So this is complicated, and it would require, you know, there's a whole other workshop, basically. But I see that Yunsuk still has his hand up, and I think that he may have also an input on the question of what happens if you try to publish an upload, say, paper or something like that on the worm. Yunsuk, I think you're muted if you're trying to speak. I still can't hear anything. It looks like your mute button is still on. And that's Dr. Yunsuk Cho, Professor Yunsuk Cho from Texas A&M, who is a very frequent guest of ours, actually. But it looks like he's having some audio trouble, even though he had his hand up before. So maybe we move on to another question first, and we can come back. We can always steer back to the strategy question of what is a smart thing to do.
Oh, hello. I guess I fixed it.
Okay.
Right. So, well, I'm not sure about this publishing the upload type of paper. But I guess there was one interesting thought that I put together while listening to Dr. Whittemore's presentation. So you talk about backup and restore, which I guess is very valuable. So maybe that's a good way to sell this. And also you mentioned just briefly about the work of Stellark. I guess you're talking about some artistic aspect. But if you, for the general audience, Stellark is an artist who does a lot of interesting body augmentation work, like adding a third arm and then trying to control it with some other means. And he was able to show that the human brain is actually quite adaptable so that it can actually control these augmented body parts. And also there was a recent study using the third thumb. Maybe you have seen it or not. So some people came up with a very interesting contraption. You add a third thumb that is controlled by two Bluetooth devices. So you can flex it, and also you can rotate the thumb. So it's on the opposite side of the hand. So now people can use it to grab stuff, large stuff, and do all sorts of things. And there was a recent study, a brain imaging study, that showed that the brain representation actually changes in response to this newly added thumb. Right? So it's a kind of a body augmentation. So with this flexibility of the brain, combined together with this idea of backup and storage, you can think about not just adding a third body part, but let's say you add a small brain, like augmented brain, and then allow your brain to control it. So this way, you're using your natural brain to somehow figure out how to interface with this augmented brain to maybe store and restore from that kind of augmented brain. So do you have any thoughts on that?
I hear you're getting into, the third brain. That's what I was talking about with the meta brain. And with Elon Musk's Neuralink, they're all type of they're tertiary brains. I loved your example of the sixth thumb, or the second thumb, third finger, second thumb, and how the body adapts to it. I'm currently experiencing something opposite to that. I had to have 3 toes fused together, and I'm waking up in the middle of the night with a little bit of a claustrophobia that I know I'll get over. It's just an adaptive reaction to having only 3 toes now, let's say. If 3 of my toes are fused, then they become 1 toe. So I'm trying to train my brain that it's 1 toe. So I have 3 toes. So it's all the adaptive of neurologically how we perceive these brains. But I think that you're right there, and you're really onto something. And thank you for mentioning Stellark. He is quite phenomenal. I think he's more of an engineer. He's not an artist. I mean, he comes from the art world, as many scientists and engineers do. But his experience is in AI and robotics, and certainly using his body as his tool. So are you suggesting that there be a third brain, or which is controlled by the core brain? So with an upload, that seems like that's right in par with the whole theoretical approach to uploading. Randall, do you agree?
Well, it's certainly part of what we envision that uploading is going to be about, is that you do enhance, that you do have the freedom to change yourself in one way or another. There is something rather distinct about it, though, because the augmentations are typically not something that is replacing or taking over function of the brain. So it still leaves the question of how do you actually get what is currently in your brain into some other substrate. I think sometimes there's some hand wavy stuff that gets done. People will say something like, well, if you plug in a computer long enough, then you'll exist as much there as in your normal brain. And maybe if the computer's big enough, you'll mostly exist in there. So then there's hardly anything lost when your biological brain dies. Maybe that's true. I don't know. It seems, unless you really make sure that you're taking over many of the very specialized functions that the brain has, such as working memory, which is quite different than long-term memory, then I wouldn't say for sure that that's the case. And that you somehow transfer the memories that you have in your biological brain, because these memories are laid out in the connections all across your brain. And what is making those connections be somewhere else? I mean, that is the uploading process, right? If you manage to then somehow transfer that over to this other brain, well, that's the same as what we're trying to do, right? Upload the brain by emulating what we can find in the existing brain, but you still have to analyze what's there to do that. So that's why, we tend to always come back to that first. It's kind of like every time we get asked about uploading, and someone will ask, well, what are the bodies going to be like? That's a fantastic question. We love those questions because, we love dreaming about them ourselves. It's just that there's so much to be done just on the topic of whole brain emulation itself, of getting what's in the current brain, that, that just basically eats up all of my mental space most of the time. And probably for most others here as well. Yeah. Oh yeah, that's right. You have work about that. That's right.
Yeah, I was thinking about that and your description of how, but it could be almost as seamless as that. Like right now, I'm here in Scottsdale, and you're there. And all of our colleagues and friends are in their various locations around the world, and we're communicating seamlessly. Only a little bit of, it's like had a little bit of audio, but otherwise pretty seamlessly through this. And while it's not me because I'm here, it's a communication channel of transference. And then you'll record this, and it goes back to the earliest questions in cinematography, and then the chemistry of filmmaking. Is that actor still in the film that's shown 20 years later, and even in black and white? Is that simulation of that actor really him, and are we seeing it years away? And I think that, of course not. It's a photograph that's moving of a person. The core issue that most of us have talked about for all these years has been, what is personhood, what is consciousness, and how much of our biological wetware do we need to be able to say that an uploaded whole brain emulation is that person. And, it's the same with chronic. Some people say, oh, you just need the brain and the neurological functioning within the brain. Others say, no, you need the whole central nervous system. You need this brain stem and the spinal system, which is a channel of delivery of nerves throughout the body. And, you can get into, when does life begin? Is it in the embryo? Is it in the fetus? Or, is it in, when a baby can say its first words and become self-aware? So who knows? I mean, it gets into all sorts of fuzzy philosophical, ideological areas and ethics. I think the main thing is, you just got to start doing it. And I loved your first question, Randall, is do you think, or I'm going to paraphrase something about, can we do a particular simulation of the neurological behaviors and interactions of a nematode performing a task and see what that looks like or the brain functioning. And would that be the best mimetic engineering to get people to go, oh, it can be done but it's an animal, it's a simple animal, but maybe in other animals it can be done. So I think that people, we go down all these slippery slopes, and again, and you were very correct. Consciousness can be viewed differently in different fields, but it's the same thing with taking a look at, memory can be determined differently in different fields as far as what it is and when it is when you get outside of biology, and it's all conjecture. So I think that people need to see an example, either as you described or a gamification of it, where you go in and play it and you maybe participate with it. And even Ben Gertzwell and David Hansen have come up, with Sophia. Sophia is a robot. It's just a head and a, upper torso. But Sophia got citizenship in Saudi Arabia, and no one blinked an eye when women can't even, I don't know if women can drive now, but it's been a real hard thing for a woman to be able to drive a car and be on par. But Sophia just walks in or is carried in and gets citizenship. But she became a symbol of something that was beyond gender biases or binary systems. She became a symbol of automation and robotics. And her first generation of communication wasn't highly intelligent or intellectual. And we even see that with Stellark's work with his head exhibition, where he's asking himself questions with a 40-foot head on stage. Beautiful to watch, but it becomes real. You believe that head of Stellark is part of his consciousness because it's his head and it's so big and real. And it's, again, here's that thing, aesthetic. So that whether it's the sound or the images or the presentation needs to assuage people to pay attention through that aesthetic quality. If you did a rap song on it with, fuck your mama and, blah, 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 and then had it, I think it would turn a lot of people off. So you'd have to do it in a different way, but also invites all generations, it's, so that's why I think gaming is really good. 3D modeling is really good, but something people can play with.
I think this is actually the first time that we've had the F-bomb in a workshop on Carboncopies.
I'm sorry, you can edit it out.
No,
I hope
fine.
the Federal Communications Commission bar you from showing this.
I have no idea. I mean, YouTube already behaves strangely sometimes. Like they closed one of our channels at one point for we don't know what reason. They never got back to us on why.
Well, edit that out to be on the safe side. But it is that how we approach it. You can look at something get turned on or turned off, and we're also woke these days. So you can have skinny worms, large worms, pretty worms. I'm teasing here, but.
So I know that there are actually two questions coming right up. One that I want to mention because it came in through the YouTube live stream channel. by Tony Fatica, and I thought it was a good connection with one of the things that you've already said. And then there is a question that I know Alexander McLean wants to ask. So Tony was asking, and you'll see immediately how this connects to what you were talking about before. He was asking, are there any estimates out there that say the percentage of time humans think in the past, present, and future? If past thinking is a lot, that could change the whole brain emulation strategy versus present or future. So I see the connection here with the previous discussion in, you were mentioning something about which parts of the brain do we need to be careful to emulate, like which aspects of what the brain does. And I think what he's kind of pointing out is the same sort of question. It's sort of the success criteria for whole brain emulation. Depending on what people care about or do a lot, some things may be more important, maybe more essential for working emulation than another. So that's, I think that ties in definitely with the whole question about augmentation and all that. I'm not sure exactly if it also ties in with worm emulation. Maybe we're kind of on a different topic right now.
I think it ties in. You have to edit this down and maneuver a little bit, but if one of the issues that would help finance whole brain emulation at whatever level or whatever project or sub-project is that it really addresses a core issue of humanity today in all societies, and that is memory loss, dementia, the rise of early onset Alzheimer's, and the urgency of this within the biomedical field, and the urgency of it with the in the augmented prosthetic field, and looking at how to use applications on your smart devices to help you remember things and whatnot. As far as the working memory, it's important for, when you're writing a paper or, designing something and engineering something, and even creative thinking to keep your thoughts so that you can reuse them. But when you're thinking about what memories people, as they get older or have levels of cognitive de-enfranchisement, would be the long-term memories are what we remember over time. And I know with my mom, with her dementia, she would talk about things that I was like, oh God, and it would be the same thing over and over again, but it dealt with the past. So you don't want to get stuck in that rabbit hole. We don't need all of our old memories, but we need the ones that we care about. So there you get into more of an emotional thing. So it'd be interesting to look at the brain under the processes of the brain of where we show an emotional hippocampus reaction to a memory. That's good. And maybe a couple bad ones, to remind us about not sticking our hands in the fire, but not all good memories will make us great people. The bad ones help steer us in better directions. But the continuity of the neurons, and I want to remember what your question at the beginning of the conversation. So that's important. So the working memory is pretty darn important for sanity's sake.
Yeah. By the way, I see that Dr. Stephen Larson managed to join us now, which is fantastic. Welcome, Stephen. I know that Alexander still has his question up here, which I will read out in a moment. But first, I'd love to just jump back to the first thing that I was asking, because I wanted to ask both you and Natasha, and Natasha's had a chance to answer, and others have commented. My question was basically, given what you've already achieved, and the command line model that you have available and all of that, in terms of whether it would help or not help move forward the big project, do you think that it would be, like, what would happen if somebody, say, took that or you guys, of course, took that and published it as, say, here is a first emulation of the worm brain, it's a draft, we've got it, or, hey, look, this is the upload protocol for a worm, for the nematode. This is how you can go from the data to the emulated brain. I know that sounds a little bit sensational, to put it that way. So that's exactly my question is, what happens if you use that sort of here, look, for this tiny animal, we can do it at least to this degree. Wouldn't it be wonderful if we could do this for something bigger? Would that be a net positive or a net negative, or something in between? What do you think?
Is your question mainly related to an announcement of a certain kind rather than the existence of the code base?
Yeah, right. It's more a question of how to frame what you have, right? Because you could basically present things any way you want to, right? Yeah, so it is really about that. It's a framing question.
Yeah, well, it's just probably one of those things where, you know, one looks at one's own work and sees all the caveats, right? And other people look at one's work and perhaps see more in it than than you yourself do, right? So I am proud of what we've achieved and what that bundle of code represents so far. We've, I think, made references to it in our 2018 paper in the Philosophical Transactions of the Royal Society, but haven't really announced it in the way that you're speaking about.
Right.
Also because, I don't know, it's sort of an interdisciplinary accomplishment, and the standards of multiple disciplines, you know, start to apply. So, for example, if it's being judged from a neuroscience perspective, you're going to want to draw some specific insights from what you're getting from the model that are relevant to and tie back to the literature, say for, you know, C. elegans neuroscience, for example. And that requires, you know, some amount of interpretation. But, you know, you make a point though. I mean, there's other disciplines where that's not necessarily the standard that one needs to publish towards. And, you know, also having come out of the AI and CS world,
yep.
Yeah, there are different things that you can say about that. So, yeah, I mean, I'm open to suggestions. I've got a bit of distance from that since it's been out for a few years, and we're looking at it differently. So, I think I'd love to hear, you know, what you think and others think as well.
Yeah. I mean, I think there's a bigger conversation to be had here, but it is just interesting to compare how, you know, the natural sort of way that neuroscientists present their work and try to, you know, make sure they don't overpromise too much, even though some of us, like, say, I don't want to name too many names, but some are better at being sales people for themselves than others are. But then you look at, say, DeepMind or OpenAI, and they, you know, basically just put on huge shows to demonstrate what they've got. Like, you know, let's take on gamers and show that our thing can beat them or something like that. Yeah, they have a very different approach to publicizing what they've done. Yeah.
Indeed.
Oh, Joe, you have something related to that?
Yeah, thank you. This was something that I asked Natasha about a minute ago, but I'd like to get your take on it as well. And that is, well, I guess to put it a little bit differently, the demonstration that I'm kind of waiting for is something where we show that we can reproduce the behavior of an individual nematode rather than sort of an amalgamated generic nematode. So I'm imagining something like we train a set of worms to do some behavior, you know, swim towards the light because there's always food that way, or whatever, climb a chemical gradient. What it might be, anything we can train the worms to do. And then we scan in their connectomes and configure our simulation and show that the simulated worms show the same behavior. And I'm wondering what you think of that idea and how far away is it? How realistic is that?
Yeah, well, the good news is the data collection for that exists now. We are, the field, I suppose, is getting pretty good at automated segmentation from video of worms, and even at a large number of them on a plate, segmenting them all out and then following the trajectory of dozens and hundreds of worms as they, you know, do different things on a dish is definitely doable right now. So using that as your sort of prior data set, I think is doable. I think that what starts to get interesting is obviously not just reproducing behavior that a worm has done, but predicting behavior that an individual worm will do. That starts to get very interesting, and it does lead to some, I'd say, open questions just in the study of behavior, right? Which is, you always qualify, predicting behavior with sort of large categories of things. So for example, some of the observations of worm behavior have started to classify behaviors into sort of large groups, like, simple things you can think about, like, is it going to crawl forward or is it going to crawl backwards? Because those are very discretely different, behaviors. But, then turning, to the right, turning to the left, are behaviors. And then, of course, there's other behaviors related to sort of reproduction and digestion and egg laying and these sorts of things. How frequently, does this worm lay an egg? How frequently does it, get hungry and orient itself towards food? So when you talk about predicting an individual, there's always this question of, well, to what resolution, right? And how precisely, and are we talking about to the millisecond, to the microsecond, right? It's obviously more and more difficult the smaller the time frame is. But the purpose of the cell-by-cell reconstruction of the worm is to be able to do that. It's just that having dived into it for a while, we now appreciate even more than we did at the beginning how difficult it is, and what additional data we're going to need to bring to bear to do it appropriately. So.
Do you mind if I follow up, Randall?
Go ahead.
I think maybe it's sort of the central neuroscience question is, when a worm learns some acquired behavior, how is that encoded? And are we even capturing it when we, do our electron microscope sections? Or is it stored in some, chemical way, or, methylation of genes or something that is entirely invisible to our instruments? And so a demonstration that the simulated worm, shows those same acquired behaviors would be proof that we're capturing the right data. And conversely, if we find we can't do that, it might indicate that we're missing something important.
Yeah, Joe, you just brought up something very important, and this is work that I know some individuals who are working on parts of this, but it'd be really interesting to see a simulation of the chemical codes interacting. And even though there could be a chart that identifies what those chemical codes mean, but you could have one simulation showing what it looks like, the visual of the firing of neurons and movement, electrical charges, and another one simultaneously showing what chemical input and output is causing it, or it's arriving at. I think it'd be beautiful to look at and fascinating, because the first one draws you in as a marketing approach. And the second one offers the scientific viability that is clearly understood and is, known within the scientific realm as far as the organism's chemistry.
I love those ideas.
Yeah, thank you. That's fantastic. Let me just quickly try to merge a secondary set of questions that has been in our chat right now. It started with a question that Alexander McLean had. It was a question about nanomedicine. And he says, it seems that the majority of us are in agreement that nanomedicine would likely be essential in making whole brain emulation possible. I guess I'd love a definition of nanomedicine first too, though. The sooner it arrives, the sooner whole brain emulation arrives. I've noticed that there hasn't been much news about any progress in nanomedicine. Are you aware of anything new in that area? Is it an area that the whole brain emulation community should invest time and energy to help make it happen? Or should we take a hands-off approach and let it just gradually happen whenever it does? And I guess, Natasha, if you want to repeat what you said to Alexander, that's fine, or I could just read it out. It's whichever you prefer.
Oh, either or. And why don't you go and read it? Because I have Robert's book in my lap right now. He gave it to me for my 50th birthday some years ago. And I'm going to read his definition since it is his baby. So why don't you read my response to Alexander, and I'll be quickly looking for Robert Friedis's definition of nanomedicine.
Okay, great. So Natasha responded, excellent question, Alexander. You're spot on. Nanotechnology was on fire in the early 1990s and AI as well. But AI moved forward and nano was left a bit behind it. However, there are biomedical research areas that claim to be doing nanomedicine that are not actually doing what was envisioned, with the nanocomputers being injected into the body to repair cells, etc. However, at least there are some steps towards the larger goal. Nanomedicine here is actually looking at molecular biology. Unless there is a robot computer involved in the tiny nanorobot system, it is not really nanomedicine as Robert Freitas first introduced the concept. To which Alexander then, I guess Alexander agreed and said that concerning, since it's, a lot of time has gone by since then, progress just hasn't really been keeping on going. So if you'd like to give us the original definition.
Oh, yeah, I'll just check it. Can you hear me? Because I'm kind of leaning back in my chair.
Yeah.
Okay, so in the foreword by Eric Drexler, Eric explains what nanomedicine is, and I'll just give you that short sentence from Eric. The term nanotechnology is used to describe a variety of nanoscale technology. More precise for present purposes is molecular nanotechnology, the ability to construct objects with atomic scale control. In lay terms, this is called building atom by atom. Okay, so Rob Friedis built off of Eric Drexler's concept of nanotechnology in his book, Engines of Creation, and his doctorate at MIT, and thought, well, if we can move atoms and move molecules atom by atom, we ought to be able to move cells and go inside the cells and deal with the proteins and the different chemical functionality of the cell. But I'm looking and I don't unfortunately see a specific definition in his book, but his book is fantastic. If anyone wants to read it, it's called Nanomedicine by Rob Friedis. It's one thing to remind us that not everyone knows what we're talking about to define the terms up front.
Yeah, and there has, sorry, thank you. There's also been, there's been a lot of movement in the terms of what the terminology is applied to as
here it is.
As you were pointing out.
Okay. About nanomedicine, molecular blah, blah, blah. Right. Okay. Nanomedicine, here, this is a quote. Nanomedicine is the preservation and improvement of human health using molecular tools and molecular knowledge of the body. Well, that doesn't really help. That's kind of more theoretical, isn't it? And I think that Alexander gets to the issue here of why don't we have it, and that maybe is a red flag that we don't have it because people probably don't understand it clearly enough. Oh. And bottom line, it's tiny computers, tiny robots that are on a nanoscale that can be injected like a vaccine or like an eye drop or a pill into the body and are programmed much like more finesse than stem cells, of course, but programmed to actually go to a specific area of the body and do repair work to clean up any messy senescent cells and get rid of them, and to look at any molecular damage and repair it. So the whole point is, instead of atom by atom, it's molecule by molecule or cell by cell repairing the damage that causes senescence and aging.
Yeah, I think it's in terms of where definitions have gone. You can hear from the definition there or the description there that it can be a bit broad and vague, like saying, something that operates on the molecular level and is based on knowledge of the molecules and molecular processes in the human body. That sounds a lot like pharmaceuticals and human body, medicine treating issues with biomedical processes in the body. So then this whole idea of you take a pill and it does something, well, that's also kind of pharmaceuticals, with the difference being that, the more, transhuman vision of it was really something that you can directly program to do whatever you want, and it will go in and it will make the changes that you want to see done. And maybe it's even self-replicating, so it can just keep going. But again, the similarities with biology are so there. It's like your immune system cleaning up old cells is also very similar to that. So I think that's why a lot of the people that I would talk to today, when they talk about nanomedicine, they're really thinking not so much in terms of what Robert Freitas was doing or is doing, but also in terms of synthetic biology and things like, what George Church is doing with all his DNA robots and that sort of thing. It doesn't have to be something made of silicon. It could be something made of carbon.
You made an interesting point here in mentioning Church's work, but prior to that, when you were mentioning, in pharmacology goes in and alters the chemical composition. But what's beautiful is, again, going back to the visualization of these things, most people wouldn't know what proteinomics is or what's inside DNA or inside a cell unless they had seen these beautifully interpreted graphic 3D modeling or gamification of it, where they can see it in actually functioning, and they go, oh, that's a protein, and that protein almost looks like a character walking across a strand of cells to get to deliver its message, because the whole body, the whole system is delivering messages throughout the whole body, and they're messengers, and so we can anthropomorphize them, and that's a good thing, because they are part of us, so they can be anthropomorphized. And it really helps people understand better.
Yeah, that's true. And I mean, in the end, it won't matter whether it's, synthetic biology or something else, as long as you can work at that scale. I think that right now, if you imagine what would you do to carry out a whole brain emulation procedure, like you want to collect all the data that you know about the brain, about the connectome, about the strength of the synapses and all that. Well, there certainly have been models where you would try to use molecular tagging to get the connectome, to get information about the connectome. Those exist. That is an attempt to go in that direction. Overall, though, the tools available today to do synthetic medicine or synthetic biology are still they're in the early phase. It's like if we go back decades in where computers and semiconductors and that whole thing was still in the early phases. And that's why most of the work that you would do today, if you're trying to say make a neuroprosthetic for say the hippocampus or something like that, you're not going to wait for those tools to catch up. You're trying to do something a little different. And even for Robert, when I last talked to him, this was actually a few years ago, unfortunately, we were doing due diligence on his company at the time. He wanted to be funded, I believe, by Brian Johnson's fund. And we were asking him about the sort of things they were trying to do, because they ultimately wanted to get to self-replicating nanomachines. But they were trying, of course, to build everything at large scale first. So it was more an external approach, using atomic force microscopes to move molecules around and atoms around. So in that sense, it's more like what you would see in a in a chip factory, where you're building your integrated circuits with large machinery on the outside, instead of having them construct themselves on the spot. So it's just interesting to see the parallels there, and also the development. So I think it'll get there eventually. It's just a little while
I think that it makes sense that AI came first in the early 90s, when we would debate and discuss which is going to come first, AI or nanotechnology. AI needed to come first, because you can't do nanotechnology without AI. And then robotics has really been growing over the past decades. So that's exceedingly important to nanotechnology, because they're nanorobots. So maybe these other fields have to do their work and build public awareness and support and education. So it doesn't seem like a horrible thing or a bad thing, but that robots can be a good thing, and AI can be a good thing. Otherwise, nanomedicine could have been, more Frankenstein-esque. So I think that maybe it's okay that nanomedicine didn't come about quite yet, but I hope it gets here before, while Rob is still highly active in his field. I think, like Eric with nanotechnology, Eric, this is more simpatico of me, but Eric deserves to be, recognized again. And, so. Hopefully. And I think that nanomedicine also from an environmental stance, if anyone is interested in that, nanomedicine certainly offers incredible alternatives to the breakdown of the ozone, or, air pollution or oil spills in water, and recycling. It's very much needed for a future world of not only awareness about any type of environment issues that are damaging in the ecological system, but to all life.
Yep, yeah, that's probably true. I want to take this opportunity, because Stephen managed to join us, to ask some of the questions that I had hoped to ask a little earlier, just because they were sort of immediate follow-ups from his talk. So I was wondering, first of all, a very practical question. Right now, if we could get more people to install this Docker container and run your current combination of bests from the Openworm project and give feedback, do you think that would be an effective way to help move the project forward for the people at Openworm, or are you already overloaded with things that still need to be improved?
I think it would be fascinating to be able to talk to more folks about it. We are also revising our documentation currently, so feedback on where it's lacking right now and where people get stuck right now, I think would be great. So yes, folks installing the container, and then having the a mindset and approach that's always valuable in an open source project, which is to continue to refine what they find rather than sort of picking it up, getting stuck, and then going away. Would be greatly appreciated. Yeah, it'd be greatly appreciated to continue to hear about, like, okay, well, so I got this far and then I didn't understand, and then here's how you could improve your documentation to make it even more clear to me, like, what was going on. I think would be good. I mean, I think it's not terrible documentation, but folks with different backgrounds are probably going to come to it.
That's awesome, because I think maybe particularly the people who are already involved with our BrainGenix project, they're doing work on a simulation platform and more. They would probably be interested just to try it out, just to install, see how it works, compare with what they're trying to do. So that's the interesting avenue. Yeah. And just from what you've learned working with C. elegans, if you were in charge of transitioning that work from the problem of C. elegans and that brain to building neural emulations of mammalian brain circuits from connectome data, let's say, if you just imagine you're in charge of that project right now, you're in charge of the mouse connectome project, or taking that to the next level, how would you go about that, given what you've learned about methodology? I know it's a huge question.
Indeed. Well, you'll have to forgive me because I'm not up to speed on exactly where all these projects are. But I think a connection to behavior is critical, right? And I know that some of these efforts have envisioned that, and some of them have even gone into building behavioral setups for these sorts of things. But I do think it's essential. It's one of the reasons we attacked the worm in particular, because behavior was so accessible. I think we need to have that in these other projects, because the central nervous system is there to coordinate behavior and activity in the world. The good news is that in the last 5 years, these algorithms for computer vision for parsing out behavior of rodents and animals in general are getting a lot better. And so it's actually a lot more practical now to be able to get pretty precise recordings of behavior from individuals over time. And if you can pair that with recordings, of course, and then combine the dynamic activity with the structure, that's of course the combo that you want to be having for actually going from structure to function.
Mm-hmm. Yep. And I guess in terms of the target animal, because right now, there are groups that are really interested in the mouse and groups that are only interested in human work because they want to do something that immediately helps patients. There are people who don't want to work with mammals. They'll work with, say, Drosophila first, or with zebrafish. Is there a species that you would say would be the best next target? And I mentioned the zebrafish on purpose because, you know, there's that whole function and connectome thing that you've got from the nematode. You can sort of see that there as well. Would you bet on one of them as the next great sort of platform animal?
You know, I'm still of the belief that because these model organisms differ in scale and each of them have structures that are analogous to humans, that they all have a different story to tell that contributes to the understanding of the human brain. So just the fact that, you know, C. elegans lacks several important scales of organization that you find in humans, so it allows you to focus on things more like the fact that the neurotransmitters are the same, and focus on sort of cellular and circuit questions before going up to systems questions. So I'd say my perspective is that, you know, we could have important discoveries in all of the above, really, that contribute at different levels. So you learn different things. So you learn like cortex specific things in, you know, rodents, but it might be a lot harder to come up with sort of principles of circuits things in rodents because there's so many neurons to deal with. But boiling that down to smaller organisms, I think you can get different kinds of lessons. So that's how I think about it.
Mm-hmm. Yeah, that's very systematic. I see here that actually that Anita Fowler had an interesting question for you as well. Anita, do you just want to ask it directly yourself?
Sure, I think I can do that. So earlier you referenced being able to potentially look up the ladder. And I'm wondering if you think you're at the stage where you're beginning to see how the algorithms being observed might be improved on or changed to make the organism function more efficiently or function like a different organism even. And if so, would you mind elaborating or explaining what you meant by the ladder? And if not, how close or far away do you think that is? And would you say that gaining perspective is a matter of simple learning or improving methodology?
Thanks for your question. I'm not exactly sure which response you're referencing, but say a few things, and if I'm answering your question, let me know, and if I'm not, let me know.
Awesome.
So I think that I look at nervous systems as byproducts of an evolutionary process that had to solve the basic problem of keeping the organism that it was attached to alive. And so the minimum standard for that nervous system was to process information enough to allow that organism in its ecological niche to survive. When you look at something simple like the C. elegans, this becomes very clear. And from that perspective, you know, with 302 neurons and able, you know, and a species that's very abundant on the planet, you start to realize that, wow, so all the neurons that, you know, we've got in human brains, you know, it's adding things for sure, but it's doing additional articulations on top of that primary mission. So when we talk about like algorithms that we want to understand, what I've been very interested in is, okay, there's that information processing problem that evolution has solved. It solved it in really small brains with very few neurons. And then on top of that, we've built these other things. So can we understand the principles of those algorithms at the level of small brains? Right now, it's very difficult because we're analyzing tissue that is, that our best way into it is to focus on the biophysics of that tissue. You know, when we're looking at the neurons, we're looking at what physics it has. And it's a complex, nonlinear, dynamical system. And so all the work to do simulations is to have our computers help us deal with the complex dynamical system that we've got there. But I think fundamentally, that dynamical system is implementing some kind of algorithm that we can understand independently of those complex dynamics once we do enough merging of data and simulation so that we can see what that is. So this idea of going up the ladder again, right? And again, I'm not sure exactly what you're referring to, but the way I'm thinking about it is unpacking that fundamental information processing step in a small animal, and then understanding how that algorithm could be further and further refined and specialized to still meet the mission of keeping the animal alive, but doing it in ways that allow for something like humans, where, you know, we occupy now a much bigger ecological niche because we're very adaptable, and but our brains are still maybe fundamentally, you know, carrying out that same algorithm. Obviously, it's doing it with a lot more neurons, and so the dynamics have probably changed a lot to carry out, you know, that algorithm. And I don't mean to necessarily suggest there is only one single algorithm, quote unquote, but I think this is the mindset that I'm approaching it with. That may, you know, be a profitable way to look at it. So I'll just say one more thing, and I'm talking a lot here, but, you know, what I'd love to see us get to at some point in the future of neuroscience is to be able to take these connectomes that we are investing so much effort and technology into getting, and be able to translate it into an information processing block of some kind, so that we could look at that tissue and say, okay, yep, this is implementing a blah, you know, a blah function. It's probably going to be more abstract than, you know, this is the place where, you know, the kind of cognitive functions is going to be more abstract than that. But it's also probably going to be different than things from the computational world that we're familiar with, like this is an ALU, like an ALU or a CPU or that kind of thing. So it's somewhere in between those extremes. I'm hoping that we can find things at this level that's small, and then start to identify in tissue, brain tissue in other animals, patterns that we can reuse. So there we go. I don't know if I answered your question or just talked for a long time, but there you go.
No, that was great. I'll definitely try to process it a little more and maybe have a follow-up later. Thank you.
Great. Natasha says that there's a great link that we can also use. So that's in the chat, I think. 3 top contenders for research on their behaviors and connectome. Okay,
I put that there because Stephen was so articulate in explaining and answering questions. And I was thinking about the zebrafish and what would be the advantage. And it was one of your earlier questions to us, Randall. What is the difference between using the fruit fly, for example, or some other less complex animal like the zebrafish and C. elegans? The zebrafish shares 70% of its genes with the human, and that's kind of a 2 thumbs up. And I think it's somewhere around 85% of the diseases shared with humans, and that's wow. But each one of these 3 animals has a different, contribution to scientific research. And I think that I still go back to the C. elegans seem to be the simplest, and maybe start simple, keep it simple. And whereas the zebrafish is, a stunning animal as well. And the fact that it does share so many of its genes with the human is a big plus. So there'd be some push and pull there. But it's interesting to take a look at these 3 top contenders. Sorry, I have a typo in my post, but the 3 top contenders for what could be significant differences in building out a connectome relationship between the neurological functioning and behavior.
Thank you. Yeah, that's true. And I think that this link is great. We just have to figure out a good way to make all of the stuff we're collecting here available to everyone.
I know. I don't have access to the other.
No, I know. We had a short discussion about that just before this all started. Kind of, we should make, we should make a list of like everybody's name who was mentioned, all the names that we've been dropping and all that sort of stuff. So we'll come up with a way to do that. One of the things I've noticed when people talk about the right animal model to choose is that, at least in the circles that I've been working in the last few months, the conversation always goes back to what tools are available in those animals. So for example, can we easily make knockouts for certain things we're trying to look at? Or is there optogenetics available? And then in the cases like the zebrafish larvae, hey, they're transparent, so we can do calcium imaging of the entire brain at the same time, and we can do, the connectome, electron microscopes as well, so we can map those two on top of one another. So those sort of things keep coming up. But one of the very good points that was made in a recent conversation there was that, there's a reason the tools exist. It's because there was a great interest in that animal, and then the tools were developed. So it doesn't mean you have to get stuck with the animals where these tools are already available. You can say, hey, this is a great target animal to work with, and then build the tools for that if there's enough grant money to make that happen. So that's just a little rant about that. I know that we still have questions that have actually been collected and that we didn't have any discussion about so far. So maybe it's a good time to give those people a shot as well. I see here that Lennon, you've got a few questions up. Did you want to mention them yourself, or shall I read out your questions?
You can read them out.
Okay, I'll do that then. So, is it clear in each of the questions whether it's a question for Natasha, for all of us, or for Stephen? Quick check.
I think I responded to Stephen's first and then Natasha, but I'm welcome to anyone's input, of course.
Okay, fantastic. So then I guess Lennon's first question is, based on what we've learned from nematodes regarding a representational and algorithmic account of their neural behavior, what conclusions can we draw about higher order processes that might exist in mammals? Such an example might be generating I think it says literary metaphor, or trying to create a dance that represents an idea. If nothing can be gleaned at this current time, what steps should be taken? So I think Lennon is looking for a connection to higher order cognitive processes from what we've learned in nematodes. And I believe this question was aimed at Stephen.
It's a meaty question. I love it. I'd say, first of all, we haven't solved it in the worm yet, right? So it's a little early for retrospectives on what we've learned. But in terms of where it might be headed, I think kind of harp on this idea of understanding the algorithm that small brains adopt in order to solve the information processing problem, and then thinking about how that information processing problem might, how that algorithm could be used as a pattern in larger brains, right, and in human brains. And so the link between cognitive processes, as I think about it, is a whole lot of patterns of processing information that got, that worked out really well, that got duplicated over time, that we packed a lot of it in. Obviously, you look at cortex, and the fact that cortex is wrinkled as it is, and essentially like a sheet of paper that got, jammed into the skull and packed in there, suggests that the pattern of cortex is one that, it seemed like the more that you had of it, the better the species got, right? But if you're unpacking cortex and you're looking at the circuit, and of course there's lots of work going on there to understand what the cortical circuit is doing. I think what I'm most interested in is, what's the underlying sort of math and computation problem that is analogous between these two things, because, it's neurons in the cortex, and it's neurons arranged in circuits that are giving rise to these cognitive processes, and it's neurons that are bundled up in a nerve ring in the worm, doing the totality of sort of the processing of the worm's behavior. So if you can sort of say like, well, okay, what are the things that both of these animals, have to do? So information has to come in from outside, and it has to hang around long enough inside the nervous system to then be transformed in some way, right? Some sort of transformation has to happen on those inputs. And the transformation generally needs to turn into appropriate behavioral output, right? So when you start going through, different kinds of neural network literature to try to find candidate algorithms that you might use to try to sort of, by squinting at them, apply them back to the nervous system. For me, I've come across reservoir computing. So I'm looking a lot at that and trying to draw analogies between what we see in reservoir computing, which is a little out of date now, actually. So it's like kind of got passed over in AI by deep learning, because deep learning was sort of more efficient for certain things. But I think reservoir computing holds some better analogies to what neurons have to do. And this is not my independent original idea. You can actually go back to Markram papers in 2002 when he was, it was part of the genesis of reservoir computing was looking at actual cortex and trying to make analogies there. But I actually think there's still something there for us to draw connections between them. So what I'd say is that if there's a transportable metaphor between what we're learning in C. elegans and cognitive processes, it's to get real clear on how do bundles of neurons compute when their input is, on the one hand, all of sensory experience, and their output is all of behavioral experience in something like the C. elegans. And in a little chunk of cortex, the input is this highly processed information coming from other areas of cortex and brain, and the output is also very high level, abstract signals to other parts of brain that then eventually end up boiling down to, okay, I'm going to move a muscle in a particular way.
Yep. So yeah, it's fundamental. It's information about fundamental computations, how they're being carried out, sort of computation that can be done in these neural systems. Quickly, for people who don't know the term, how would you describe reservoir computing?
It's wow. I think I'm so deep into it that the basic definition is something I'm still looking for. So everybody can Google it, but it's a kind of neural network, okay, that starts off by building what's known as a reservoir. It can be implemented in lots of different ways. It can be implemented with a network. So it's a recurrent neural network. Can be implemented with excitatory and inhibitory units mostly, but it's at equilibrium. It's sort of at rest, and the best metaphor that I have to it, it's like a liquid that like say the surface of a pond, and you throw a pebble into it, and then it starts reverberating. Okay, and reservoir computing, this recurrent neural network has to satisfy a couple of properties in terms of the way that it resonates. One of the properties has to do with the fact that it's memoryless, so that after it's done resonating, it goes back to a state where you can't recover the signals anymore. The other is that there is a period where that resonance doesn't immediately decay, like instantaneously. So it has to resonate for some period of time. I know I'm butchering the formal definition. And then from there, you have readout units. And those readout units can be essentially linear readouts from the reservoir. So the surprising thing that happens is that you can put in complex time-varying signals into the reservoir, and just with linear outputs, you can train up any arbitrary function that you want, linear or nonlinear. And this has been shown in a couple of papers I can throw you guys. So what's really appealing about it for biology is that it allows you to imagine neural systems that evolution could reasonably create kind of through random mutation. Because it's not very hard to have a bunch of neurons that are recurrently connected, right? And it's not that hard to have, some linear readouts that are synapsing through chemical synapses. So if those are the two main requirements, and generally there's like some amount of activity that needs to ring or resonate through the reservoir, evolution can go to town on that pattern all the time, and just by maybe adding some more neurons to the reservoir here or reducing them there, you can amplify or attenuate certain kinds of signals that are coming in to allow for a greater amount of computation. The last idea that's important for reservoir computing is just the idea of taking lower dimensional signals, blowing them up into a higher dimensional space, and then using the linear readouts to the way that the computation can happen is that these linear readouts are able to essentially define subspaces in that high dimensional space where they can pull the information out. So for me, it's a intuitive way of being able to get a grip on parsing through with very complicated data and very complicated anatomy to try to bundle essentially the interneurons of the C. elegans, the sort of 70 to 120 in the nerve ring, and kind of see if it fits to call that a kind of a reservoir. And then to look at the motor system of the C. elegans that drives the muscles of the body as these linear readout neurons. And so that's kind of the mapping that that I'm exploring.
Thank you. That's great. And I think that reservoir computing is going to come back, especially because it's driven from computational neuroscience. It makes a lot of sense to look at it, and we see very similar models emerging, for example, in research on working memory and also on context generation in dentate gyrus. So those are some areas where the reservoir computing model, in addition to memory, is also being applied as a tool, at least. I think we're going to see that come back again. I'd like to now switch over to a question for Natasha, which is also from Lennon. And it's actually on a somewhat different topic, but it completely makes sense to ask this now since we have you here, Natasha. Lennon is asking, what are the observed effects on behavior of repeated, if you know this, repeated cryonic exposure to a simple organism? Has anyone bothered to repeatedly thaw and refreeze, if we want to call it that, organisms and observe the results? And Michael Ulrich immediately added to that, how do humans compare for multiple revivals? So I know you may not be able to answer this, it's just questions are out there.
Okay, so as I'm understanding this, the question is, can you take, or has there been a C. elegans or a set of C. elegans that have been put in vitrification or cryopreservation with a cryoprotectant and revived, and then put back into the protocol and tested to see the integrity of their functionality after the second go at it. It could have happened and I might not have noticed it. When you're working with hundreds of worms, and Stephen certainly can understand this, it's like trying to identify a neuron and how many times will it light up based on this stimulation. It depends on other behaviors going on. So I think that might answer that question. But I could have, and not knowing it, because but I think the process I used is used by Ramon Risco in Spain, and that's where and he patented the straw. You pull up a single C. elegans, or maybe two, but we were doing single ones in a straw, pull it up, hold it in that straw, and then place them into the liquid nitrogens individually, and then put them, in the warming bath after X number of hours or minutes, however, the protocol determined, and then put them back in the Petri dish. It could have been done by mistake, when going into the dish, maybe one got put back into the dish and then was brought back up. So I wouldn't be surprised if that was true. I cannot verify, I cannot confirm or deny whether it survived, because some of them didn't. And the way you can tell that they didn't survive is they become straight as a stick, or some of them aren't very healthy and then they wobble around kind of disoriented, and it's really sad to watch that. But I think the simple answer is I don't think so, not that I know of. And the second part of the question was about the complex animal of a human. How would it compare to being tested numerous times?
Multiple revivals there, yeah.
I guess one of the issues here, and it'd be like pharmacology. Like I got hives really bad, and today I'm not scratching. I had surgeries over this pandemic, and I had a terrible reaction to the anti-inflammatory. I mean, the anti whatever it's called thing. Any of the medicine. Antibiotic and the anti-inflammatory, anyway, on my body, driving me crazy. And I'm going, why am I allergic? Why is it going to my toe and my right finger, my thumb, and then up in the top of my head and the lower part of my back or the inside of my knee? What is causing the nerves, or why is it going in those directions? It's kind of like that. You don't know. It's so complex. And I'm sure if someone could put a tracker on it, as Stephen suggested, track some of these connections and relationships, it'd be easier to do. Since there has been no human that has been put in chronics and revived, I can't answer that question. But going back to the comparison to pharmacology, the drugs that we put in our body can cause reactions. So if you're using an cryoprotectant, if you're using a cryoprotectant, which has to be used at this stage in vitrification or cryobiology, then there could be some adverse effects as to how long it takes that to leave the system, and doing it multiple times. But now let's fast forward to the future. In the future, when we have nanomedicine and our ability to realize uploading or whole brain emulation, it'll probably be a non-issue, because we would just wipe that out. So I think that's a far future answer. But and here we are in June of 2021, there's no answer, and I apologize.
That's totally fine, of course. Although I'm sure that everyone who's hoping to take advantage of cryonics next year and to be thawed the year after and then put back in again for a year because they just want to.
It's still the best plan B. It's still the best plan B. And I think that the only competition would be with, uploading, but I still like plan B and having the choice to upload, cross-load, and download.
I'm going to switch back again to Stephen now because I see that Michael, was it Michael? Oops, I just suddenly lost it. Yeah, that Michael had a question for Stephen that, did you want to ask it yourself? Go ahead.
Yeah, thank you for the question. I think it's a great one. And you know, I like to think of projects that are open and open science really as jumping off points for lots of folks to do, I think, all of the above next steps that you're talking about, should it be helpful. I think one of the reasons we started the project was an observation that in areas of physics, there was a much more productive conversation between theorists and experimentalists. Okay, so looking at the discovery of the Higgs boson, right? And if you guys saw that great documentary about that one, I don't remember the name of it right now, but this really profitable conversation between the theoretical physicists who were predicting that the Higgs boson would, I don't know, have this particular signature, and the debate and the discussion there, and then they had to go and test it, right? Biology is, because it involves all these nonlinear dynamics, and because it's hard to just write, the one equation of a cell, right? Cells don't really give themselves up to being described in single equations, at least for most of the processes we care about, right? It's kind of only simulation in which we can, particle fever, yeah, thank you. It's only within simulation that we can start to do the same stuff that the theoretical physicists do. And I think astronomy is also another place where modeling is very productive. So to answer your question though about what are next steps, I really see that my hope is that Openworm would begin to inspire folks to be able to have that kind of dialogue between experimental biologists and computational and theoretical biologists, computational neuroscientists. I'm particularly interested in neuroscience, but the C. elegans, the majority of the labs in C. elegans aren't focused on neuroscience. They're focused on other interesting problems, fundamental, molecular and cellular mechanisms of life, right? There's research on how fat gets produced in adipose cells, and aging research is a big area in C. elegans, right? So, but there still is not in biology as much of this idea that there is a really tight conversation between the folks that are imagining how biological processes might work and might come together with the folks that are actually, doing tour de force experiments using amazing genetic molecular imaging techniques that are coming out right now. So realizing that it's hard, in larger animals, part of it was that to do it for, humans, you got to do a lot more. Like the computational problem is just a lot harder because there's a lot more state variables, if you will, to track, like a lot more neurons, and therefore every neuron, a lot more molecules, and therefore a lot more synapses. And so it's a harder problem to bite off. The idea of starting with something small and then attaching into that community a really profitable conversation between theoretical work and experimental work, and then essentially having that idea be picked up by other areas of biology was kind of the idea. So I think the model could keep going for a long time is kind of what I'm saying here. The same way that, we're not done figuring out theoretical physics or particle physics, the same way that we're not done understanding the universe, but what we have in those areas is, sort of the tradition of building models and having conversations where experimentalists trust models and believe in models. But in biology, it's been a lot harder to do that.
Thank you. That's wonderful. And now I'd like to either read out, or maybe Joe wants to say it himself. He has a very practical question related to C. elegans work.
Yeah, this is just something I've been wondering. The most recent paper I could find that was about work producing fresh C. elegans connectomes. This was Stephen Cook in 2019. They were still tracing the EM images by hand. And I'm wondering if you know what might be the holdup with applying automated segmentation like they're doing in the Drosophila and mouse connectome research, applying those same automated methods to C. elegans.
Steve Cook's a friend and collaborator. And so I know a little bit about some of those, conversations. But the last that I, remember, I think is just purely a matter of these communities are very small, and folks that do the automated segmentation, they're very new, and the algorithms are newish, I guess, 2018, 2019, a few years ago. I think it's more a matter of just the particular collaborations not yet being there between. So it kind of makes more sense right now, or folks have been more excited, I guess, to do Drosophila, because I think there's this idea that because Drosophila hasn't been done, and that's where you want to apply the algorithms, whereas, C. elegans has been done, quote unquote, a few times, although I think, you want to be doing it at a much high throughput way and doing it across lots of individuals and comparing against them. That's another subject. But I think it's just the collaboration hasn't been put in place, to my knowledge, to apply them yet. But, a paper could come out next month, and I wouldn't be too surprised to see that actually they have been applying those algorithms to C. elegans as well.
Okay, thank you.
Fantastic. I think that we've gone through most of the questions that we had already collected. Oh, there's one more coming right in. So, Brigitte, either you can say it yourself directly, or maybe you want me to read it out.
No, I can say it because I've had this question for a while. When are glia going to be included in the connectomes and in these projects? Because they're a very important part of behavior as well.
Yep. Speaking not as an experimentalist, I suppose it's a little difficult for me to project. I don't know if Natasha has a better perspective than I do on that.
Oh, forgive me. And Stephen, thank you for tapping me on the shoulder. Could you please give me the question again, and I'll see if I do have an answer or not?
Yeah, do you happen to know like when glia will be included in these projects? Because they're a very important part, of behavior.
Absolutely. Oh, what a great question, and I can't answer. But I love that you asked it, because it inclusivity. Let's bring it in. Yeah. So I don't know. Sorry, I can't.
That's fine, of course. I mean, we can't know the answers to everything right now, but so also, I don't know anything really about C. elegans, which is why I cannot comment on glia in C. elegans. I know that there is obviously a lot of attention being paid to glia right now in mammal brains, at least in terms of trying to find out in what way they participate in the computation that's going on that's of cognitive importance. So are they, we're definitely past the point where, where neuroscientists think that glial cells are just there as like a sort of a life support function for neurons. There's more than that. There's certainly an involvement in modulation. The question of how much actual computation goes on, where you would say like every individual glial cell has an important purpose. I mean, we can't even necessarily say that every individual neuron has an important purpose. That is, as far as I know, still outstanding, so it's not entirely clear. What I think does matter is that when you look at it as a computational system, that even when we talk about how glial cells are included in the computation, that it's mostly a discussion of, well, how does what the glial cells are doing, how does it affect the firing of the neurons? Like, how does it affect whether they are more sensitive to firing, or whether they will fire more rapidly, whether they can fire a stronger burst. So in the end, it's still about, okay, how does it affect what is going on in the neural communication? And so in that sense, it's a little bit similar to discussions about, okay, well, how much attention should we pay to these diffuse messengers, type of neurotransmitters, rather than, say, the direct transmission at the synapse. And in the end, you, as a modeler, you can go in and look and say, well, okay, sure, a diffuse transmission is something that happens over a certain distance. You can kind of give it a kind of an R² type of equation, and you can say, well, that messenger connects with the neuron somehow. It causes a reaction in a receptor, and then that will activate or change the membrane potential, and potentially therefore when you have a spike. So you can again translate it into that, so that in the end we're always taking all of this biology and we're stripping away the meaning of the particular biological component, and we're trying to turn it into, hey, we could label these messengers whatever we want. We could make glutamate do what AMPA does right now if we just flip around what the receptors mean as well. So in the end, it's not about those particulars of that biological bit. It's always about what effect does it have on the computation in the end. And so far from what I've seen, the discussion of what effect does it have on the computation in the end always still comes back to how does it affect how the neurons in this region that are encoding for this are affecting the neurons in that region that are encoding for that. So it's sort of participatory. That isn't saying very much. I know I'm leaving out a lot here.
But Randall, I think you said it beautifully. The whole time you're talking, I'm thinking about homeostasis, feedback and control. And we're talking about AI here with biology, and the glia are like the regulators that control the system. And so they're not pawns, they're incredibly important to keep the equilibrium, the safety net of that system. And in cybernetics, the feedback and control by Norbert Weiner's, in looking at the homeostatic system, it's so essential for every system. So, and the other analogy I was thinking about outside of cybernetics is, when we found the gene and, looked at the chromosome, it was an aha moment. Okay, the gene, yes. And then we went, okay, now in the gene is the protein, and proteinomics is now a very important field of study. So it wasn't just the gene, it was the inner workings, as you said, the relationship and all the variables of the system make the system.
Yeah, that said, though, I know that there is actual work looking into, well, how much specific computation goes on in glial networks. And it's just that to me, it still feels like it's I guess it's still not a very well substantiated field yet, but that's just me. And yes, Stephen, please, your take.
Sure, yeah. I mean, I will tell you one of the reasons it's hard to do glial research in C. elegans is the same reason that it's very difficult to do sort of single cell patch clamping in C. elegans. It's a much smaller system. I know of only one lab, the lab of Miriam Goodman at Stanford, that trains scientists to patch individual cells. And of course, patching cells has been a way in which we've understood the effects that glial cells have on neurons. You can even patch glial cells, or you can use indicators that will show you how much neurotransmitter is present around a synaptic cleft, and then see how much maybe a glial cell is soaking up, that kind of thing. But there is just the same tools that we've used in, say, slice experiments in rodents are harder to apply just sort of mechanically to C. elegans, and so that's why it's difficult. And again, there's very few labs that do that kind of work. But I would hope that we could apply the same tools that we've used in larger organisms and go back down to the worm and understand them, because there is an inventory of some of the glial cells, some of the support cells for the nervous system that are present. It's not like we don't know what they are, but I don't know of almost any papers that have looked at their function relative to the neurons in C. elegans.
Right. So I actually have looked at the glia in C. elegans in the male tail.
Oh,
yeah.
Brilliant.
Yeah.
Awesome. Great.
And
So
I
buried the lead there.
Yeah, I used GCamp. That's the tool that I used. And GCamp does work well in C. elegans glia. So.
Good. Yeah, an optical technique. It makes sense. Yeah.
Right.
Yeah.
Yeah, no, the patch clamping and stuff is so hard in C. elegans because the cells are so small. They're just so much smaller.
Exactly.
So actually, you may be the best person to answer the question that you asked. What's your take on it?
Well, I would like, the people who do all the connectome work on neurons to do glia at the same time. because that would be very helpful for me as a person who does a lot of the GCamp work in the neurons and in the glia to figure out, okay, what cells are these actually connecting to? Like, is this glia that I'm seeing firing at the same time as a neuron? Is it just talking to that neuron? Going back to your question, is that everything seems to be neuronal computation? Or is that glia actually talking to the muscles that are doing the work? I don't know. At this point, I don't know. All I know is that I can tell you that glia is firing. I can tell you it's releasing dopamine, but I can't tell you, is it just impacting the neurons or is it actually directly communicating to the muscles that are producing the behavior at this point?
Okay, so this is a great request for people doing connectomics research. Yeah.
Well, the thing about C. elegans is that the micrographs, the electron micrographs, are out there, and yes. I would imagine that the membranes of the glia come up just like the membranes of the neurons do, and I think people have segmented them out. So if the question is like, can they be included in the structural understanding of the cells of the C. elegans, I think the answer would be yeah, for sure.
Yeah, I've definitely seen them do it, some of it in the papers that have been published, which is very helpful to me. But yeah, it's definitely something to highlight that would be useful for the field to answer, to essentially to really answer the question, what exactly are glia doing, right? Like, where are they in this computational process? Are they just with the neurons, or do they actually provide some of the information directly to the output system as well?
Yeah. What role should we look at them as having?
Yeah. Right.
So,
there is one thing here I think that just bears mentioning, which is the idea of timescales. So from what I've pulled together about glial cells in other animals, the role that glia seem to be suggested to be playing out are on the order of seconds to minutes, whereas neurons in terms of firing are happening more at the millisecond timescale. So when we think about simulation, we think about our simulations when they only involve neurons likely being a lot more valid in those small timescales, but then getting increasing amount of error for lack of things like glial processes and other cellular processes as you go beyond the millisecond time range into seconds. So I think
that's a fantastic point, because mode switches are really important, and where glial cells affect that, I think that's very relevant. That has to be put into any system model of how any brain, C. elegans or another brain, works. And that's actually a place where there's a little bit of an analogy again between the glial cells and diffuse messengers, which they also tend to act on a somewhat longer time scale, and also are involved again in upregulating, downregulating, mode switching, and so forth. Definitely not something to be ignored. Yeah. So let's see. Do we have more questions? Because I know we're coming on to what we normally sort of call the end of a workshop. So we have about 10 minutes there. But I would love it if anyone has any pressing questions for our guests, or just, for everyone. If this were the all hands meeting, we'd say it's open floor time.
Well, I'll jump in with one more. And actually, the question about glia brought this to mind. And I'm wondering, Stephen, in the Openworm simulations, what are you simulating besides the neurons? Are you simulating the whole animal on a cell-by-cell basis, or is the rest of the animal more of an abstract model?
Yeah, great question. So it's sort of a system of simulations where we've connected a couple things together. So there's the neuronal simulation, but then there's also this body simulation where it's not every cell. We've sort of abstracted the body into a set of particles that represent largely the tissues of the cuticle of the worm and the muscles of the worm. So, and there is a 3D visualization of this, and you'll get a movie of that that will be on your computer if you download the Docker container and run it, because it will compute. So basically, the idea is that, and then also as part of that simulation, that body is inside of a environment with a either a liquid or a gel environment that is configurable. So that the body is bumping up against particles that represent a liquid or a gel, and that gel pushes back on the cuticle, which gives the opportunity for sensory input to come back in. In our latest version, we haven't yet got the full closed loop of the sensory information impacting the right sensory neurons, but the other way is working in terms of the motor side outputting. And causing the cuticle to bend as the appropriate muscles are contracting and expanding. So that's a big part of the model as well.
Great, thank you.
Anyone else at this point? Looks like we are getting to a close here. Good. Then maybe I'll just start wrapping up, and then Bill will close everything off for us. First of all, I just want to thank both Stephen and Natasha for the amount of time you have put into both the interviews and the time here at this workshop. I know it's it's valuable time. And we're really thankful that you were able to join us and give us your insights. So that's certainly, and then of course everyone else who joined in and asked questions and helped make the event possible. I think that that we certainly have learned something here about where C. elegans work really fits in with our road mapping, at least, of how whole brain emulation is going to happen. And I still think that, that just the process of combining both function and structure, as you've been able to go and do in C. elegans, is something that, because that's still such a very new process, it's a very new thing to do, it's something that's going to give us a lot of insight into how to go about a project like that, even if we're just dealing with a very small circuit, like say, or we're doing retina or something in the human, or maybe beginning to analyze some of the subsets of networks that are actually in that data set that the Allen Institute just collected from that cubic millimeter of mouse brain, or the hemibrain at Genalia of the Drosophila. These are all going to be very useful. And we shouldn't underestimate the importance of worms, of course. I know no one's mentioned it here. Everyone's just kind of been tiptoeing around the big issue here, but as we all know from anyone who's a big fan of Dune, worms are important. They are not called the maker for nothing. So we should be grateful that that work is out there. So I know I'm being silly. It's that time, we spend too much time in these meetings, and then I get silly because I get tired. So anyway, I'd like to say thank you. And Bill, please feel free to take us into our closing.
I'll start with our usual closing procedure and list all the announcements and etc. First, let me just share my screen. Okay, we've reached the conclusion of the June 2021 workshop event. Thank you all to our speakers, our panel experts, and our audience, as well as our volunteering staff, patrons, and donors. Thank you all very much for making things like this possible. Just one more reminder that we are a 501c3 nonprofit that exists and operates with the sole mission of advancing research towards whole brain emulation. As such, we appreciate the many contributions in the form of donations and volunteering time that have allowed us to organize events such as these. If you want to become a monthly donor or make a one-time donation, you can visit our page at carboncopies.org/donate. You can donate by PayPal, credit card, bank transfer, or cryptocurrency. You can also contribute by volunteering as a team member. We're always looking for motivated people with any level of experience or any type of experience. If you have a technical or scientific background, you can join our active research projects, technical review, or roadmap updating. If you have experience with activities in nonprofits, then you could help us recruit, coordinate events, maintain expert networks, raise funds, reach out and educate, etc. And to find out how, go to our page at carboncopies.org/openings and connect. You can also sign up to our newsletter or follow us on our social media accounts. Our Facebook group can be reached at facebook.com/groups/carboncopies. We also have a Reddit page, r/carboncopies, and we have a Twitter account. Our handle is @carboncopies.org. Thank you all very much, and hopefully we'll see you again at our next event.
Thanks, everyone. Have a wonderful day.
Thank you.